particular ORF5, which is designated as C1, which pushes the bacteriophage to go for the lysogenic pathway. And again, you have ORF6, which codes for protein, which pushes the phage towards the lytic pathway. Now, apart from that, what I found is a very, very interesting gene, ORF7, which codes for antirepressor, which basically codes for antirepressor, the lytic pathway. Or whatever it decides, whether it decides to go for lytic or lysogenic pathway, in case it decides to go for the lytic pathway, then there are two important proteins in the bacteriophage which must actually function. One of it is gene 53, which is also referred to as endolysin, and the other is gene 52, which is also referred to as choline. So today, my entire talk will focus on this gene 52 and gene 53 and how together they form the bacteriophage's ultimate weapon, the choline endolysin system. Now, just to give you a brief idea about how choline and endolysin together lyse the bacterial cell and allows the bacteriophage to come out of the cell, I've made a small animation. So say this is the bacterial cell wall where you see the peptidoglycan layer, and after the peptidoglycan layer lies the cytoplasmic membrane. Now, if the bacteriophage has infected the bacteria, then the choline proteins which I have told you, GP52, these are going to go and insert themselves in the cytoplasmic membrane like this. Now, within the cell, there are also the phage and anything. They are waiting for the choline to make holes in the cell, in the cytoplasmic membrane. So at a particular concentration of choline, it forms holes or it makes punctures in the cytoplasmic membrane of bacteria. And now the endolysins, which were already produced by the bacteriophage, will cross the cytoplasmic membrane and reach the peptidoglycan layer and will make holes or rather rupture the peptidoglycan layer, through which the bacteriophage can actually come out of the bacteria. Now the question is, why is the choline or the endolysin system so interesting to us? It is interesting because they are basically bacteriolytic in nature. In the sense, they have the capacity to kill the bacterial cells, and hence they can be used as alternative strategies against antibiotics. When antibiotics are failing, we can actually try to use the choline endolysin system. But before we decide that GP52 functions as a choline and GP53 as an endolysin, we need to study them very minutely if we want to actually, take them forward as an antibacterial agent. GP52 really function like a choline, like lytic protein, and how does its structural domains contribute to this activity? So if you see here, this is the basic structure of GP52, which is annotated as choline, and it has a very interesting structure where you can see that the first 12 amino acids in its N-terminal end, they are highly positively charged. Now this is followed by a transmembrane domain TM1 and then a second transmembrane domain TM2, and these transmembrane domains are actually going to insert the protein within the bacterial membrane. And the 12 amino acid long N-terminal tail, this is also, we are expecting it to be very important because it is so highly positively charged. So next what we did, in order to understand whether, which domain has which function, we created a series of mutants of GP52, N-terminal deletion mutants, where we first removed this 12 amino acids in the N-terminal, and we, this particular mutant was designated as GP52 delta 1 to 12. In the next mutant, we removed the transmembrane domain 2, and we named it as GP52 delta 1 to 35. And finally, we removed the transmembrane domain 2, and we had the protein, which we call GP52 delta 1 to 64. Now, after creating these series of mutants, we wanted to see how each mutant functions in the host cell. And how did we do this? We did it by turbidity reduction assay. So we found turbidity growth curve of the cell in which we are overexpressing GP52. And what do we see here? When the wild type GP52 is overexpressed, there is a sharp decline in the OD of the cells, which is indicated of very, very strong lytic activity. Then what we did, we took the other two mutants, that is delta 1 to 12 and delta 1 to 35, and then you can see here, this is delta 1 to 12 and this is delta 1 to 35, and you can clearly see that the decrease in OD is not as as rapid as we had seen it in case of the wild type. Here there is a slight OD decrease. So partial activity of the protein is retained, but it is not as effective as the wild type. So this indicates that both the N-terminal 12 amino acid chain as well as the transmembrane domain have a slight role to play in the lytic activity of the protein. But the most interesting observation was here. Here what we had done, we had removed the N-terminal tail, the transmembrane domain 1 as well as transmembrane domain 2. And what we found is, in case of this mutant, there is no drop in OD, there is no decline in OD. It grows just as sharply as the wild type, indicating a complete loss of lysis in the choline protein. So turbidity assay actually reveals that the transmembrane domain 2 is essential for the lytic activity of GP52. So this was, our first hint that the entire choline protein has domains which modulate its functional activity. Now, next, so here we actually concluded that TM2 deletion abolishes GP52's lytic activity, and it is critical for the membrane disruption. Also, TM2 likely forms part of the pore or structural scaffold, which is very, very essential for lysis. Now, what was our next step? We wanted to see if this wild-type protein or its mutants would actually, affect the membrane permeability of the host cell, and this we did by using the beta-galactosidase release assay. So what we did here is, so you can see here clearly, these are normal BL21 cells which have no choline in them, and you can see there is no release in beta-gal over a period of 0 minute, 30 minutes, 60 minutes or 90 minutes. There's hardly any release of beta-galactosidase. But when you look at the wild-type choline, you can see that with time, there's a huge increase in the release of cytoplasmic beta-galactosidase. And this is only possible when the membrane is disrupted. So this gives us a clue that the wild-type choline, when it gets overexpressed in the bacterial cell, it actually disrupts the membrane. When we look here, this is the mutant where the N-terminal 12 amino acids have been removed, and here you can see there is a dip in the release of the cytoplasmic beta-galactosidase, indicating loss of lysis in these cells. And then this is the other mutant where the TM1 domain has also been removed, and we can see that here also there is a decline in the release of beta-gal. But if we compare it with normal BL21 cells, these are of course higher. Now the interesting result was in the last mutant where all the N-terminal domain as well as the transmembrane domain 1 and the transmembrane domain 2 were actually removed. And here we can see that there is absolutely no beta-gal release. It is as negligible as in the wild type, as in the normal BL21 cells. So the results, what does it confirm? That wild type GP52, there is a strong beta-galactosidase release, as we can see here, indicating robust pore formation. Also, delta 1 to 12, that is this mutant, and delta 1 to 35, that is this mutant, here we can see there is only partial release of the enzyme, indicating lesser membrane disruption as compared to the wild type cell. And finally, when we look into the last mutant, that is delta 1 to 62, we find that the release of beta-galactosidase is almost negligible, almost like the normal BA21 cells, indicating that if we express delta 1 to 261, where all the transmembrane domains as well as the N-terminal stretch of level 12 amino acids are removed, the membrane remains intact, and that is why possibly we do not get any beta-gal release here. So TN2 may be, so here we concluded that TN2 may be essential for pore formation and lytic activity of GP52. So what do we interpret here? TN2 is essential for membrane permeabilization, and it supports a model where TN2 forms the core structural element which drives GP52 mediated lysis. These findings align with choline-like functional characteristics where other proteins have also been studied. However, these assays only tell us what happens, but it does not tell us where or how GP52 behaves inside the cell during the process of making membrane punctures. To explore how GP52 behaves within cells over time, we fused it to a GFP molecule, that is green fluorescent protein, in order to visualize its location within the cell by confocal microscopy. So here you can see this is normal BL21 cells expressing GFP, where you can see GFP throughout the cytoplasm of the cell. But if you look closely here, this is basically the wild type GP52 cells, and you can see that at the end of 30 minutes, the fluorescence is not throughout the cytoplasm, as you can see here. It is all throughout the cytoplasm. But here it is basically lining on the membrane. You can see here it is lining on the membrane. Okay, and at the end of 90 minutes, if you look this side, then you can clearly see that the cells are actually the GFP is actually migrating to the poles, and since the GFP is fused to GP52, it is indicative of the fact that GP52 is also migrating to the poles of the cells, unlike the normal BA21 cells. So this was a very, very interesting data for us because this was very different from many of the other rulings which have already been studied. Other than, I think, R.Y. Young, who has shown terminal localization of choline proteins, no other cholines have been reported to show this terminal movement. So this visual proof reinforced that the N-terminal transmembrane domain are the proteins at this tag, guiding it to the membrane where it must act. So once we were, just give me 1 minute. Yeah. So GP52 undergoes temporal relocalization during the expression. This is something which we understood during this particular work. Now, we wanted to see the functional dissection of N-terminal and the TM domains. Now, if you remember here, all the mutants that we have studied have removal of the first N-terminal 12 amino acids. Then it is the first N-terminal 12 amino acids along with the transmembrane domain 1. And finally, it is a combination of the N-terminal 12 amino acids, the transmembrane domain 1 as well as transmembrane domain 2. However, now we wanted to look into specific TM domains. If we remove only TM1, then what happens? If we remove only TM2, then what happens? So this is what we wanted to study, and we generated mutants just like that. So we generated mutants lacking TM1 only, TM2 only, and both TM1 and TM2. And now we wanted to see how, the effect of the protein might actually change. So here you can see this one is the uninduced culture, of course. This one is the mutant in which the TM1 has been removed. And this one is the mutant, the green colored curve is the mutant where TM2 has been removed. And the light blue colored curve is the one in which both TM1 and TM2 has been removed. And what we understand here is the TM2, just give me 1 minute. So here what we can see is the TM2 deletion actually abolishes the lytic activity. confirming its critical role. Because as we can already see that when TM1 is removed but TM2 is retained, the cell still carries out very efficiently the lytic activity of the protein. However, when we remove TM2 in this case or in this case, the lytic activity is completely lost. So this actually gave us some important clues. One, delta TM1 gives good turbidity reaction, but lytic activity is majorly retained. Delta TM2, there is minor turbidity decrease with complete loss of lytic function. And where you have delta TM1 and TM2, there is no activity, and it is very similar to the uninduced cells. So we conclude that TM1 contributes but is not essential for the lytic activity of choline, and TM2 is actually indispensable. If you remove TM2, as in this case, the complete lytic function of the protein is lost. So it's possibly the core membrane disruption domain of the protein. So this functional data actually aligns with our beta-gal release assays, reinforcing this particular model. So TM2 plays a central role in GP52 mediated lysis. So far, we have seen the functional evidence that GP52 disrupts the bacterial membrane, causing cell lysis and permeability changes. Now, to confirm and quantify the impact of GP52 on membrane integrity, we performed a membrane potential assay using DISC. So, normally what we know is from Holine's data that Holine basically disrupts the PMF or the proton motive force of the cytoplasmic membrane. And now we wanted to know if GP52 also does that. Does it have any impact on the PMF of the cell? How we can actually study this by using a dye known as DISC3. And what this DISC3 basically does is, it basically, if the cell, if the PMF of the cell is compromised, the DISC3 is going to get released from the cell, which we can measure by fluorescence. So we did this, and what we found is, this is of course, the normal cells where you do not see any fluorescence. This is GP52 where, only TM1 is released. This is GP52 where both TM1 and TM2 are deleted. This is the wild type GP52 where you can actually see a large increase in the fluorescence, indicating that, the PMF has been dissipated in the cells. And this is the GP52 where the TM2 mutant has been removed. And here also we can see the release of the DISC3 from the cell in the form of fluorescence. So this indicates that choline not only forms holes but possibly sensitizes the cell to endolysin activity by dissipating the PMF. Now here one interesting thing which I need to tell you is, if the PMF of the cell is not dissipated, then endolysin cannot function efficiently. So we conclude that possibly choline actually dissipates the PMF as is indicated by these fluorescent bars, and it makes the cell more vulnerable to endolysin action. So we have found a combination of choline and endolysin which can be effectively used to disrupt the cell membrane of the pathogens and actually become lytic in nature. So expression of cholines alone without endolysin in bacteria is also lethal, as we can see here, confirming their ability to dissipate the PMF. So this was all about the N-terminal domain of the choline protein, what the N-terminal domain does, and how it actually leads to lysis of the bacterial cell. But here again, we wanted to see what the C-terminal region of the choline protein does to the host cell. So what we did, we have seen that the N-terminal region and transmembrane domains, the key role is in lytic activity. But the C-terminal domain has been very less studied. So here we generated some C-terminal truncation mutants, quite large mutants, and we assessed the lytic activity using multiple assays as we had done in case of the N-terminal domain. And here, as you can see, so this is the wild type, this is the normal BL21 cells, okay? And this is, of course, the C-terminal mutants, one with a 20 amino acid deletion and one with 40 amino acid deletion. And what we can see here is even removal of 70 amino acids from the C-terminal end does not disrupt the activity of the protein. So this indicates that the C-terminal domain of the protein must be involved in some other activity, but definitely not in the lytic activity of protein. So here are key observations. Even large deletions such as C70 retain turbidity reduction assay, and this suggests a non-linear relationship between the C-terminal length and the lysis function. So this became very clear to us, and we concluded that GP52 acts as a rhodine-like lytic protein with the N-terminal, TM1 and TM2 domains driving membrane disruption, and the C-terminus possibly fine-tuning the lytic activity. Together, these regions enable coordinated membrane targeting and bacterial lysis. Now, other than GP52, which is the rhodine, I have told you that there is another protein which is important for the lytic function of the bacteriophage, and that is basically the GP53 protein or the endolysin. We call it the endolysin. And what we see here is, it is the enzymatic enforcer. So we actually purified the endolysin protein, as you can see here, and we, we purified it by affinity chromatography. This is the STS page, which shows the thick, pure blend of endolysin. And we did a zymogram assay, as you can see here. What does a zymogram assay do? So in the gel, you have the target for the endolysin, that is the peptidoglycan layer. And wherever the endolysin is now moving in the gel, it will create clear zones, as you can see. Okay, so this basically indicates that our GP53, which is the endolysin, is very potent against Staphylococcus aureus. We did a few other assays, so you can see these are the control cells, and these are the cells which express endolysin, and we can see that very quite a good amount of killing can be observed in the cells which are treated with endolysin. We also did a scanning electron microscopy to properly see if any change in the membrane is done by the endolysin. And what we can see here is these are the normal cells. The moment the cells are treated with endolysin, you can see there are hardly any cells. Most of the cells are dead. You can see the debris. But most importantly, what you can see is that in case of cells which are treated with endolysin, you can see that there are holes and they are not as smooth and round as the wild type cells. So this indicates that endolysin definitely has an impact on the morphology of the cells. So after this, so now what is the current view of our lab? It is the translational vision. We are turning molecular precision into therapy by using it on medical device, proteins, nanodelivery, and diagnostics. Our rationale is that rising antibiotic resistance. As you know, the antibiotic resistance is increasing day by day. So this actually is going to, this system is going to help us actually counter the antimicrobial resistance. So we can make modular antimicrobials that can be engineered for specificity. We can make medical device coatings. We are actually already developing GP53 based formulation for intrauterine devices, and we can use them as biofilm disruption agents for persistent infections. We are also moving towards cytotoxicity assays to see if it has any impact on mammalian cell lines. So we are also developing some nanocarrier platforms for controlled and localized release of choline and endolysin. Enhancing the stability, penetration, and therapeutic precision is also underway. And one more interesting thing which we are doing is we are creating diagnostics for pathogen detection using the cell wall binding domain of endolysin. So the cell wall binding domain will be used as the biosensing element, and it will help in the rapid detection of target pathogens. And if we integrate it with a biosensor and surface detection platforms, then it will be one of the easiest diagnostic systems to actually identify the pathogenic bacteria in any sort of surface. So our next steps, we would be moving towards cytotoxicity testing, where we will check the impact of choline embolizing system on mammalian cell lines. We will go for in vivo validation to see the efficacy and the biodistribution of the enzyme system. And we will of course go for formulation optimization. Now for this we are teaming up with our pharmacy department, which is going to help us in formulation optimization. This is my lab. So these four people are currently working in my lab, and they are the ones who have actually done the entire work. We have recently published in a journal known as PEPS Letters, where we have got a cover image on this particular protein. And so thank you so much for your attention. If there are any questions, please ask me.
Hello. Gracious thanks to Dr. Malvika Biswas for her illuminating address. Our heartfelt gratitude to you, ma'am. Hello.
Can you hear me?
Yes, ma'am.
Yes. So I'm done with my presentation. So if there are any questions, I would be very happy to take.
Now the session is open for question and answer.
Hello. Hello. Malvika. Hello. Can you hear me? Hello. Malvika, can you hear me?
Yes, I can hear you.
Yes, I am Dr. Deshmukh.
Hello, sir.
Hello. So, I am very happy to see you here, and I would have been more happy from Goa if you would have been here.
Yes, sir, I wanted to come, but because of some other commitments, I'm a little stuck because classes have started here.
Okay.
And since it was the start of the semester, I was not able to get any leave.
Yes.
I'm so sorry I could not come. It would have been really nice to meet you all.
Okay. Your presentation was very good.
Thank you, sir. Thank you so much.
And if there is any question from the audience, they can come here.
Yes.
One question is there, Malvika.
Yes. Please ask me. Hello?
Dr. Malvika,
Yes. Please tell me.
excellent presentation.
Thank you.
This really helps in understanding why phage become a lysogeny and also why there are failures.
Yes. Thank you.
And is it possible to develop any primers for this, Malvika, so that a lot of phages, it is very difficult to do a whole genome sequencing to understand. Is it anywhere possible to develop a primer where we can identify the phages are moving towards a lysogeny? Thank you for the excellent presentation.
Thank you so much. Actually, the main drivers of lysogeny are the C1 protein. So, but we need to see computationally how much, conservation is there in these sequences. If you find a high level of conservation, which does not change with different phages, then definitely we can design a primer. But as of now, what we do in the labs, we simply do plaque assay to see what kind of plaques we get. If they are clear, then we know it is lysis, and if it is sort of cloudy, then we conclude that it is possibly lysogeny. But primers till now have not been developed.
Thank you, ma'am. Gracious thanks to Dr. Malvika Biswas for her illuminating address. Our heartfelt gratitude to you, ma'am, for the session.
Thank you so much.
I request Dr. Aparna Patade to kindly felicitate the chair. Okay. Participants, settle into your seats for awardee function beginning soon.
I request Dr. A.M. Deshmukh, Dr. G.R. Pathade. I request all the participants, delegates, students to gather into the Jupiter Hall. We will shortly begin with the awardee session. We have eminent different people who have worked efficiently into the fields of life sciences, maybe biotechnology, microbiology, zoology, plant sciences, animal sciences. As the theme of the conference is, we are integrating the modern science into translational research. These people are contributing their work into translational research and then making it possible into the society for the benefits. So the presence of all these awardees today have graced our conference, and their interaction with the students and all the participants will surely make the conference a great success. I request all the other participants standing outside the Jupiter Hall, please join us for the awardee function. The students of Krishna Institute of Science and Technology waiting outside the auditorium, please gather in the auditorium. I request Dr. A.M. Deshmukh, Dr. Shirley George, Dr. Anjana Singh, Dr. Amrita Ranjan, Dr. Ulrich Burke, Dr. Snehal Masurkar, and Dr. G.R. Pathade to be seated on the dais.
Excellence is not an act, but a habit. True recognition belongs to those who strive for perfection in their chosen field. Good morning and a heartfelt namaskar to all the distinguished delegates, eminent scientists, researchers, academicians, awardees, and luminaries gathered at the Jupiter Hall of Krishna Institute of Science and Technology, Krishna Vidyapeeth, deemed to be University, Karat. We are privileged to welcome you to the awardee function of the 3 days international conference Biomake in India 3 2026, a momentous celebration organized by Krishna Institute of Science and Technology in proud collaboration with the prestigious Microbiology Society India. Today, here we honor researchers whose groundbreaking work bridges ancient wisdom with cutting-edge microbiology and biotechnology for humanity's sustainable future. We stand in profound gratitude for the gracious presence on our dais, the international scientists and visionary leaders who will witness and bless these well-deserved recognitions. Following our ancient tradition of invoking divine wisdom, I request all our esteemed guests on our stage to participate in the sacred lamp lighting ceremony, seeking Ma Saraswati's blessing for the success of this awardee function. To lead us, I humbly request Dr. Aparna Pathade, Associate Professor KIST, to grace us with the beautiful Saraswati Vandana.
जय जगदीश्वरी देवी सरस्वती, जय जगदीश्वरी देवी सरस्वती, नमनमिदं तव शिवपदकमले, जय जगदीश्वरी देवी सरस्वती, त्वमसि निधानं सकलकलानां, त्वमसि निधानं सकलकलानां, त्वमसि निधानं सर्वार्थानां, त्वमसि निधानं सर्वार्थानां, निर्झरिणी त्वं चातुर्याणां, निर्झरिणी त्वं चातुर्याणां, दन्तरुचा शारद शशि विमले, जय जगदीश्वरी देवी सरस्वती, नमनमिदं तव शिवपदकमले, जनयसि वाचं चारुविलासं, जनयसि वाचं चारुविलासं, कलयसि विश्वमिदं सोल्हासं, तु कलयसि विश्वमिदं सोल्हासं, कुरुषे स्वान्ततमो विध्वंसं, कुरुषे स्वान्ततमो विध्वंसं, महिमा तव विश्वस्मिन् अखिले, जय जगदीश्वरी देवी सरस्वती, नमनमिदं तव शिवपदकमले, जय जगदीश्वरी, जय जगदीश्वरी, जय जगदीश्वरी देवी सरस्वती.
Thank you, dignitaries, and thank you, ma'am. Now I respectfully invite Dr. A.M. Deshmukh, President, Microbiology Society India, to bless us with his address.
So, I am very happy to address today's gathering. This gathering is a academicians gathering, but this is an answer to the many students which are sitting on the back benches. They are always asking one question. Sir, whether microbiology has a scope? Second question, sir, are there any jobs available in microbiology? And we are telling that don't ask for job, create job. And today is the felicitation of the great personalities, I will tell, of India, pillars of India. By their research, by using microbiology, they are creating jobs. So today, we are felicitating such personality. You see, whenever I appreciate efforts of Dr. Patadi sir. The reason is that today it is an international conference. Written is an international conference. Is it really international? On the stage, all personalities are international except Dr. Deshmukh, Masurkar and myself. And all awardees from Jharkhand, from Assam, you see, right from Rajasthan, from Hyderabad, that is Telangana, from Mangalore, Karnataka. Meaning is that from different corners of India. So it is a truly international conference. And there is a time limitation, I do understand. So we are not given a time to present themselves to the all awardees except Dr. Yadav sir. Dr. Yadav sir and I myself. Only 2 persons are youngest person in this hall. So, because again I am telling, the one who is mentally young is young. And Dr. Yadav is one of them. So we have given time only for a 3 minutes, 4 minutes presentation to Dr. Yadav. Otherwise, all other will present themselves on the first floor. On the first floor, there is a poster presentation, and I will request all attendees go and see the posters and see what miracle all these awardees are doing in India. What is the title of our conference? Biomake in India. And they are the true awardee, real pillars. So you go please, watch their innovation, watch their setup of the industry, watch what academics, through academics, they are idol of biotechnology and microbiology. So, and in presence of all these foreigners, right, means from different countries, they are here, and at their hands you will get an award. So deliberately, we have made the function in such a manner, they will give award to you. And this award will inspire all the awardees, I know. But rather than awardee, it will inspire to the all other students. They will also feel, if you can do, why I cannot? And today's India, again I am telling, Honorable Modi ji started Bio E3. What is E3? E for environment, E for economy, E for employment. So Honorable Modi ji is telling that in India only there will be bio business in the future. The economy of India will be stronger only because of the microbiology and biotechnology. And truly, all these, there are approximately 28 awardees are there, correct? Jayasri, total 27 awardees are there. So, all these awardees, we welcome all the awardees, And we are always Microbiology Society and Dean of this Allied Sciences from Krishna Institute. We are very thankful to you. And really, we will support you in the future also. Keep in touch with us, and we will witness more, because this is the beginning of your industry, your attempt. Attempt may be failed, may be successful, but we appreciate you are making an attempt to make in India, that is a Biomake in India. Thank you very much.
Thank you, sir, for your inspirational address. Embracing Aditi Devo Bhava, we honor our distinguished international and national guests. I request Dr. A.M. Deshmukh to extend warm felicitations to Honorable Dr. Shirley George, Assistant Professor Physiology, Manipal University, Malaysia. Thank you, sir. May I now request Dr. Thank you, sir. Now I request Dr. G.R. Pathade to honor Dr. Anjana Singh, former head, Central Department of Microbiology, Tribhuvan University, Kathmandu, Nepal. I request Dr. G.R. Pathade to present floral tribute to Dr. Amrita Ranjan, researcher, AMHBI, CPUT, Cape Town, SA. Now I request Dr. G.R. Pathade to extend heartfelt regards to Dr. Ulrich Berg, PhD in philosophy, former professor at university and the president of German Association for Homotherapy. Thank you, sir. Now I request Dr. Anjana Singh, former head, Central Department of Microbiology, Tribhuvan University, Nepal, to address the gathering.
Distinguished guests on the dais and guests on the floor, as well as participants, staffs of this Krishna Institute of Science and Technology, and one and all. First of all, I would like to thank for giving me an opportunity to address the August gathering here in front of you all. And I can see that this conference, that is Biomake in India, third, which has been organized for 3 days. It is an international conference in a hybrid mode as well. This conference has a diverse group of participants from all over the India, as well as from the various countries abroad. And I can see that we have been exchanging ideas and experts between us, and that is really one of the good points to enhance the science and technology in the science and technology, the subject, as well as we can see that the subject, especially the biotechnology and microbiology, has been suffering in having most of the time the jobs. And as previous speaker, Professor Dr. A.M. Deshmukh, with whom I'm really close to, and as well as because of him that I'm here. I'm really thankful to him also, that how we can develop ourselves, how the youngsters, that is the young ambassadors of science and technology, can not only go for a job seeking, but also also to develop oneself to become an entrepreneurship, or that you can also build your own kind of platform where you can use yourself, your ideas, your expertise, and the future science and technology followers. So again, not only in this, we have seen that from yesterday there are beautiful talks and mesmerizing talks which we also learned from each other. And besides that, not being the monotonous in science and technology, I can see that the participants, especially the students from various colleges, they have been quite energetic and being involved with the traditional culture of our South Asian countries and within India itself, that how you have taken the culture, the traditional culture of not only the in sense of biomaking, but also the culture itself, that how you have mixed together, and then that you have incorporated with the modern science, that how it will help in our modern science with our ancient cultural traditions in science and technology, which can be embedded with it, and then we can go forward. Besides this, this is also a kind of a forgotten science which is we have been leaving behind. So this science where we can also put forward and then mix together with our modern science, so we can helpful for the well-being of our human, well-being of the human as well as animal as well as plant health in one health system. So in this, I can see that yesterday's culture program also, it showed that the students not only being monotonous with science, they have come up with the cultural programs where they have shown the energetic form of their life, and as well as how the science can be also involved with others, and they have shown their talent. And with this, I would really thank the organizers for giving me this opportunity, and I again wish the conference to have a great success, because one more day is ahead of us. With this, I would like to end my small phrases and talk here. Thank you so much.
Thank you, ma'am, for gracing the moment. Now I welcome Dr. Amrita Ranjan, researcher, AMHBI, CPUT, Cape Town, South Africa, to address the gathering.
A very good morning to one and all, and very heartfelt thanks to Dr. Pathare, Professor Deshmukh, Dr. Snehal for inviting me as a guest of honor and giving me this opportunity so that I can talk to you all. I am coming from Cape Town, South Africa, but by heart and first of all, I'm an Indian. I have been working in many countries in Europe, and now I'm serving in South Africa, Cape Town. To be very frank, before coming to this dais, I had a speech scripted, but I really don't want to read anything for now, and I want to talk from my heart, because I see so much of students sitting here who wants to make their career in microbiology or biotechnology or the applied sciences and have so many doubts in mind. Believe me, when we were in your position, we have the same doubts. And we are coming from the time when microbiology and biotechnology was just a budding subject. Hardly few people knew it, that it exists, right? And we made a mark, and we are still trying to make a better mark. And I'm pretty sure that if you will work hard, if you'll be persistent, and if you love what you do, you will also make a mark. When I just look into this scenario, I'm just recalling, I think nearly 2 decades ago, when I was doing my PhD at IIT Guwahati, I was also awarded for my work in the area of renewable energy in one of the conferences. And now I'm on the opposite side, and I'm here to award all the little brains who are supposed to be the architect of the coming future India, right? So I'm pretty sure that after a decade or two, you will also be sitting here in the dais if you stay persistent and enjoy what you're doing. If you have doubt, it's good. It's good to ask questions, it's good to have some doubts, it's good to be nervous, but what is important is stay calm, be persistent, interact, make some good networking, study hard, and at the same time, try to be more applicative. From the very beginning, I have been working in the field of renewable energy and looking for alternate biostock for the production of value-added products. This has been my core area of research. And now, if I look back, I think whenever I see any waste, I see a resource. I see a wealth in it. My brain starts working that how can I turn this waste into wealth? So this is how I decided that this is the area I'm going to work. So I'm pretty sure when you will look into your own inner self, you will find what charms you and what makes you to enjoy your work which you are doing, and I'm pretty sure you will make really a good mark. So all my best wishes for all the bright innovators who are getting awarded today, and I'm looking forward to look all the posters, and for those who are not awarded, you will have another time, so keep working hard and good wishes to everyone. Thank you so much.
Thank you, ma'am, for your inspiration and guidance. May I now request Dr. Ulrich Burg, President, German Association for Homotherapy, to address the gathering.
Good morning, everybody. So first, I want to thank Dr. Girish Patade and Dr. Deshmukh for organizing this conference and for inviting me. Dr. Patade I know since long time, and he was very instrumental in helping to close the gap between traditional knowledge and modern science. He was one of the first scientists in India who not only said, oh, I'm interested, I will do some research. Many did. But he was one of the very first who then also actually performed the experiments, and that helps. And now the topic of this conference is Confluence of Indian Knowledge System with Modern Science for Sustainable Innovations and Human Well-Being. It is an essentially important topic because now we live in a time of, on one hand, you could say great problems, great challenges. This is true. We have this climate change, which basically it is all about pollution, overall pollution of all the spheres of the planet. The atmosphere, the soil, the water resources, or you could also say all the 5 elements, earth, water, fire, air, and ether, all are polluted. Now we have to come back. How can we do this? We go back and make use of traditional knowledge. But in our time, if we do that, we can't just say, oh, go back to the Vedas. We have to show that this Vedic knowledge is relevant for our time today. And this is, we do this by doing experiments in terms of modern science, showing that yes, that is working. For example, we have air pollution. If you think of air pollution in Delhi, every winter again, it rises to an incredible number. And if we can show, yeah, for example, you practice Agnihotra and air pollution comes down, that really helps. And this is something which then also everybody at his home can do. And scientists, so especially you as young scientists, you have a big job and a big chance also to show the world how we can get out of these old problems by making use of the traditional Vedic knowledge and show the world, okay, this works, and please take care of that. So, and that could lead to a much better society, which we have now, a society with peace, prosperity, and happiness for all. That is our game. So thank you, and this conference can be part of that endeavor. Thank you.
Thank you, sir. May I now request our distinguished chief guest, Honorable Dr. Shirley George, Assistant Professor, Physiology, Manipal University College, Malaysia, to address the gathering.
Respected Prof. Deshmukh, Prof. Patade, distinguished dignitaries on the dais, esteemed awardees, faculty members, and my dear students. It's truly an honor and privilege for me to stand before you as the chief guest for this award ceremony in the International Conference Biomake in India 3. First of all, I would like to thank Prof. Patade and Prof. Deshmukh for recognizing such respectable awardees with a lot of work which other conferences do not do. When there is a conference, I think only there are mostly paper presentations, and an award ceremony is very rare nowadays in the conferences. Just researchers are being heard and they are not awarded. Maybe first place, second place, and third place is given only for their presentation and not for the work which they really do. And I hope this award ceremony is not just a ceremonial act. It should definitely serve, and I hope it will definitely motivate the young minds who are stuck of what to do in future. It will definitely motivate you all to do your best and give your best. And conferences is always a networking. So I hope students and even us, we have a lot of networking after attending this conference from different parts of the world. I hope we connect to each other and grow together. Thank you.
Thank you, ma'am, for your motivational speech. May I now request Dr. S.R. Yadav from Shivaji University to share his view.
Dignitaries on the dais, of the dais, and student friends. First thing I should appreciate, that is the theme, confluence of Indian knowledge system with modern science for sustainable innovation and human well-being. So we all working for welfare of human being. But I additionally, as a scientist, in addition to human beings, we have to consider all other organisms also. This is what I feel, because not that only human beings are to survive on this planet. This is one thing. I congratulate all those awardees who got award in this conference, and I have heard those award lectures, only 3-4 slides each one presented, and each one has done excellent work, and I found that there is a great strength with our researchers and students in India. Another thing which I want to say, awards is a recognition of your work, but those who don't get award don't feel that, because who is the best person to judge you? You yourself is the best person to judge you. If you are contributing something better for Mother Earth, that is more than award, right? So don't feel that way also. Thing is, see, microbiology, plant sciences, and animal sciences, all these are biological sciences, and without microbiology, plants cannot survive. Without plants, animals cannot survive. So for entire world, one message is, only way by which we can grow and conquer this earth is cooperation, right? Microbes cooperate with angiosperms, angiosperms with animals, and this way it is a cooperation. Even we are the most successful organism on this planet. It is, because of what? What are your super qualities? Human being super quality. And I appreciate our Deshmukh sir, A.M. Deshmukh, it is a cooperation. Bringing people together, working together, and therefore we are able to conquer this earth. If one elephant come, one person cannot do anything. But when you join together 20 people, we can make elephant to run. And this way only we have got success in our life, and cooperation is very important. Now this is an age where interdisciplinary work is very important. We should not have walls, botany, microbiology, zoology, these are different departments. Unless we come together, even physics and other branches also, unless we come together, we cannot do meaningful work. And that way, it is very important. So, our, dignitaries have given wonderful message to all of us. I must thank organizing committee of Krishna Institute of Science and Technology and this conference, Krishna Vishwa Vidyalaya deemed to be University Karad, Society of Microbiology, because I happen to be a student of Science College Karad. My native place is Karad and this is my home. And getting recognized in your home has a great significance. It is forever, and that way I thank the organizing committee for awarding me Bio Innovator Award. I think that is the Bio Innovator Awards, and that is to recognize your contributions to the society. So I think this is my message, and one thing I should tell you, sustaining life, we have to sustain life. What can we do? Technology can make our life enjoyable, very good, comfortable, but sustaining life, remember what I am saying, sustaining life, which can do? To sustain life, sustain in the sense, I need food to eat, I need good air to breathe, and I need my good health, for which we need medicines also. These are the three things: my stomach, food, my health, and my breath. These are the three things which come from biological biodiversity: microbes, plants, and animals. And this sustain the life, and then technology makes our life very comfortable and enjoyable. So life sustainability lies with biological sciences, and 21st century is a century for biological sciences, and definitely it will make our life enjoyable in future. I mean, especially students, you are the pillars. We all senior people sitting here, we are working for coming generations, not for ourselves. So, for posterity we work, all of us senior people, and that way now we are going to hand over this responsibility to our students. I appreciate this Biomake in India. It has a great future, and as Professor M. Deshmukh has said, don't keep what job I will get. Become knowledgeable, you can do many good things to our society. With these words, I am thankful for giving me opportunity to talk here. Thank you very much.
Thank you, sir, for sharing your thoughts. May I now request the chairperson to address the gathering, Dr. G.R. Pathade.
Good morning, one and all. Good morning. Dignitaries on the dais, my beloved teacher, Dr. M. Deshmukh, with his blessings in the life we are going ahead in the academics and research. Dr. Ulichberg, to whom I am associated since 20 years, right from Ferguson, when I was in the Ferguson College, heading biotechnology, microbiology and environmental sciences. He is a man who has spread our traditional Indian knowledge. Agnihotra, the proven scientific technology in more than 80 countries since last 45 years. And I'm proud of him that in fact he has taught me how to perform Agnihotra. We are Indians. He has taught me how to chant the shlokas which are performed, chanted at the time of Agnihotra. Surya Swaha Surya Idannamama, Prajapate Swaha Prajapate Idannamama. He has taught us in Ferguson College, and from that time, in fact, he motivated us to go for traditional Indian knowledge. And with his motivation in last 20 years, I organized more than 20 conferences in traditional Indian knowledge. This is a fourth international conference. First international conference we organized in Pune with the Deccan Education College. Now it is deemed to be university. So his great contribution is there. With his guideline, one of my students completed his PhD in Agnihotra in Pune University. He completed PhD degree in Agnihotra in Pune University in spite of great resistance from our Indian academics. But fortunately, we got support from outsiders for this Agnihotra, and it was proven that it is a proven scientific technology, and then his registration was confirmed. Shirley Madam from Malaysia, she has appealed us as well as to you all that she has organized one international conference in Malaysia to happen in the March, October, and definitely Dr. Deshmukh and I myself will plan that so that more than 30-35 people from India, they will participate in that conference. Singh Madam, Anjana Madam, again we are associated for more than 20 years, right from Pune, when I was in Pune, right from 2006 to 2022 I was in Pune, Ferguson College as well as Haribhai Desai College I was there. And she is always instrumental in helping in different conferences and attending conferences and delivering motivating lectures to the students. As Yadav sir rightly told, that this planet is not given gift to the mankind only, to the human beings. We should not forget that along with human beings, other planet animals, living beings, they have got equal right from microorganisms. And we should not forget that. We are forgetting, in fact. If you forget microorganisms, sir has told what will happen. If we consider tomorrow microorganisms go on strike, what will happen? Nobody will survive on this planet. Life has originated on this planet in the form of microorganisms, single cell, and it will exterminate with microorganisms. So we should not forget the microorganisms, never underestimate the power of microorganisms, this is told by the Louis Pasteur. So we have to accommodate in future conferences all these aspects, not only well-being of human being, well-being of living beings on this planet. Biomake in India, this culture of Krishna, with the blessings of our Chancellor Sir, Dr. Sureshji Bhosle, and all the visionaries, we want to extend this every year. This is the third year. So Biomake India 3, this is third year. And I'm sure that our team, enthusiastic team, and future teams, this Biomake in India will go infinity. And this Biomake in India, which is aligned with the Vision 2047 as well as NAP 2020. The motto of this Biomake in India is student centralized. That this young generation should go ahead and stand on their own feet. We should make their feet so strong that, as Deshmukh sir has told, they should not ask for the jobs. They should create their own jobs. And in these conferences, as sir also told that it is very good that we are recognizing the entrepreneurships, so people. This is because there is communication gap. There is a lot of people they are working. There is no dearth of people in India who are working in different fields, including traditional Indian knowledge. N number of people, N number of agencies, N number of groups they are working. But communication gap is there. Through this Biomake in India, our plan is that all these groups where communication gap is there, that the gap should be removed. And all these groups should come together and should form a strong platform for different aspects of this developing science, including our traditional Indian knowledge. As sir told that this planet Earth is in danger because of our own activity. But human being is to err. Mistake to hum karte hi hai, but we have to rectify these mistakes. These can be rectified if you take care of every living being except human being. Then we will find that harmony will be there. Otherwise, that bad point which is called extermination is not far away. We know that climate change, every year conditions are worsening. This year also we have found natural calamities, maybe heavy rains and even temperature in summer too hot. All these things, it is in our hand, and it is a responsibility of every citizen of this planet. Narendra Modi, our Honorable Prime Minister, always says, Vasudhaiva Kutumbakam. We consider this whole planet as our kutumba, family. And hence everybody's responsibility is there to save this planet, this Mother Earth. And this is our Biomake in India is a small attempt that this should come up, and every year we are going to call the people who were called in previous conferences. That this particular bond of the participants and delegates should be long lasting. The entrepreneurs, the awardees who are called, we are very proud that they responded and they joined physically, because we made it compulsory. If they come physically, more motivation will be there to the students in spite of the instead of the online. So most of them, they accepted our invitation and they came physically, because our students, they want to see the new industrialists, their senior colleagues, they can say, because they also will go as a future entrepreneurs. And I am sure that these awardees, they will motivate, proactivate all these young generation members who have come who are not only from Krishna group, but they have come from different colleges. More than 20 colleges we have collaborated, 20 colleges they have collaborated in this conference. So I am sure that this event will be very fruitful, and the students basically, as I told that it is a student centralized activity, so students will be benefited. So again I open the treasure of best compliments for you all. Thank you very much. Jai Hind, Jai Bharat.
Thank you, sir, for sharing your experience and the visionary address. The moment we all await, the Biomake in India 3 award distribution ceremony. Our esteemed dignitaries will now present the exceptional researchers and innovators their awards. The first category we are having is Bio Startup Award. May I call upon stage Dr. Anil Shankar Yadav, Raut Serum India. I request Shirley ma'am to hand over the award. The awardee has made a world's first snake venom detection kit. Next we have Dr. Rajnish Bhati. Dr. Rajnish Bhati, Chairman, Arasu Prakash Innovation Private Limited, for the development and commercialization of sustainable agricultural solutions through organic fertilizers, microbial inoculants, and agro-waste-based biomanures. Next, we have Dr. Vaishali Kulkarni, KB Calls Science, for microbe-derived sustainable colorants replacing toxic dyes. Next, we have Dr. A. Anand from Thandai Hands Rover College for portable forensic kit for wildlife crime scene investigation. Dr. A. Anand. Dr. A. Anand. Next, we have Dr. Sujit Pompala, who is the founder and managing director of Ucha Herbs Private Limited. Next, we have Dr. Anuj Gupta. Anuj Gupta, who is the founder and director of Genotope. And he is receiving the award for integrated genomics platform with affordable point of care diagnostics. Next, we have Dr. Vinod Kumar CS from SS Institute for pioneering bacteriophage therapy against antimicrobial resistance. Next, we have Ms. Surbhi Ameria from Birdhara Innovations for hydroponics with biofertilizers for climate resilient farming. Next, we have Dr. Parag Saudagar, who is the founder and director of SK Biobiz Private Limited. Dr. Parag Saudagar, for development of probiotics reducing pesticide residues. Next, we have Amit Raj. Amit Raj from Tintera Naturals for development of plant-based natural colorants for sustainable textile dyeing. Dr. Arohi Kulkarni, AA Nandini Solutions for development of microbe or enzyme platform for feed, silage and oral health. Dr. Vivek Ghate, Dr. Vivek Ghate, Theraxil Healthcare Private Limited, for development of first in India Mycophyte TM 3D bioprinting therapeutics. Dr. Pinky Raj Sahu from MSME Nature's Nurture, founder and trustee Seva Dham, for development of Sanjeevani kit empowering 250 plus rural farmers and SHGs. Ms. Akanksha Kashyap, Ms. Akanksha Kashyap, founder, Region Packaging Bhopal, for fungal mycelium biodegradable patch packaging alternative. Ms. Shruti Trivedi, proprietor Shriji Biotech, for developing reducing chemotherapy side effects. Dr. Madhura Nerurkar, founder and director Kalanta Biotech Private Limited, for development of enzyme replacing chemical industrial processes. Dr. Sanjay Vayal, founder and CMD, Ishved Group Pune, for women-led global agribiotech expansion. Dr. Neelam A. Salunkhe, Director, Euchroma Research and Innovative Solutions LLP Pune, for micro hygiene and microbial safety services. Next category we have is Bio Innovator Awards. Professor Dr. S.R. Yadav. Senior Scientist, Department of Botany, Shivaji University. He has discovered 75 plus new plant species and authored Flora of Kolhapur. Dr. Girish Gajanan Poddar, Professor and Head, Department of Botany, Yashwantrao Savan, College of Science, Karad. He is a national expert on Indian grasses, authoring 21 books. Next are the special category awards. Dr. Avinash Survase, Assistant Professor, Department of Microbiology from Yashwantrao Savan Institute of Science, Satara, for nanobiotechnology patents for cancer detection. He is receiving the Bio National Young Scientist Award. Dr. Shiv Sharan Rohini Ravindra, Assistant Professor from Solapur University, for her outstanding women scientist contributions and receiving the award Women Scientist. Dr. Bharti Prakash, Associate Professor and Head, Department of Microbiology, University College, Mangalore, for generating 10 lakhs revenue via water testing consultancy. And she is receiving the Women Scientist Award. Dr. Pradipta Ranjan Rauta, Assistant Professor, Biological Sciences, AIPS University, receiving the Senior Scientist Award for 2,740 citations in cancer nanotechnology. Professor Dr. Devjani Dasgupta, a thunderous standing ovations for these extraordinary researchers whose innovations bridge ancient wisdom with modern sciences for sustainable human well-being. Thank you, dignitaries. Can be seated. Our heartfelt gratitude to Chief Guest Dr. Shirley George, Guest of Honors and Chairpersons for personally honoring the scientific luminaries. To conclude, I request Dr. Jayashree P. Nanavre, Assistant Professor KIST, to present the vote of thanks.
Good morning, one and all. On behalf of Krishna Institute of Science and Technology, Krishna Vishwa Vidyapit, deemed to be University Karad, I take this opportunity to express our sincere gratitude to all the dignitaries, guests, faculty members, and participants, awardees who have graced and supported the awardee function of 3 days International Conference Biomake in India 3. First and foremost, we extend our heartfelt gratitude to our Dr. Sureshji Bhosle, Honorable Chancellor KVV, deemed to be University Karad, for his visionary leadership, constant encouragement and unwavering support towards the academic excellence and research-driven initiatives. We express our sincere thanks to Dr. Neelam Mishra, Honorable Vice Chancellor, KVVDU, for her guidance, motivation, continuous support in fostering a vibrant academic environment. Our deep appreciation goes to Dr. Ved Prakash Mishra, Chief Advisor to Chancellor and KVVDU, for his invaluable insights and strategic guidance that continue to strengthen the academic and research ecosystem of the university. We are grateful to Dr. Praveen Shingare, Principal Advisor, Academics and Accreditation, KVVDU, for his constant encouragement and academic leadership in maintaining quality standards in higher education. We sincerely thanks to Dr. M.V. Gorpare, Registrar, KVVDU, for his administrative support and cooperation in facilitating the smooth conduct of this academic event. We extend our special thanks to our chief guest, Dr. Shirley George, Assistant Professor, Physiology, Manipal University College, Malaysia, for gracing the occasion and for her valuable presence and insights that enriched the awardee function. We also express our heartfelt appreciation to Dr. A.M. Deshmukh, President, Microbiology Society, India, for gracing the occasion as a guest of honor. We extend our sincere gratitude to our distinguished guest of honor, Dr. Anjana Singh, Dr. Amrita Ranjan, Dr. Urlich Burke, for gracing the occasion and offering their valuable remarks, which sets an inspiring and scholarly tone for the program. We gratefully acknowledge the support and cooperation extended by all the university authorities and officials for their guidance and administrative assistance throughout the event. I also express my heartfelt congratulations to all the awardees. Our heartfelt gratitude to Dr. G.R. Patade, Dean, KIST, for his visionary leadership, encouragement and continuous support in organizing this conference. We sincerely thanks to Dr. Snehal Masurkar, Dean Academics, KIST, co-convener of Biomake in India 3, for her support. We express our sincere gratitude to Dr. Dhanashri Patil, Assistant Professor, KIST, and Organizing Secretary of Biomake in India 3, and Dr. Narendra Kumar Suryanshi, Assistant Professor, the co-convener of the Biomake in India 3. For their dedicated leadership, meticulous planning and invaluable efforts in successful organization of this conference. We extend our sincere appreciation to Dr. Aparna Patade, Associate Professor, KIST, for invoking divine blessings through Saraswati Vandana. Thank you, ma'am. Our heartfelt thanks to the Master of Ceremony, Mrs. Ashwini Zadav, Assistant Professor, KIST, who conducted the proceedings. We also sincerely appreciate the efforts of the Powerhouse Department, IT Department, Photography team for their technical and logistical support, which ensured the smooth and successful conduct of the program. I would like to place on a record of our appreciation for the organizing team, teaching and non-teaching staff, as well as the research scholars, volunteers, whose enthusiastic participation and cooperation made this ceremony success. Once again, we express our sincere gratitude to everyone who contributed directly, indirectly to the successful conduct of this awardee function. With this, we conclude the awardee function. Thank you all.
Thank you, ma'am.
Sorry, one mistake again from our side, from my side. One awardee from Hyderabad. He has a great contribution also in the microbiology. He did a PhD under the Sandipta ma'am from Usmania University, Hyderabad. And by mistake, his name was deleted. So I will request him along with wife. He is here. So I request him to come on the stage and we'll give award. Yes, sir. Please come here. Come on the stage. Please. Please go. And again, one more request to all awardees. Please, you prepare your video of 7 minutes only. Everyone. I want a 7 minutes video. I will compile it and which will be uploaded on the website of microbiology. It will be circulated throughout the India, out of the India. Because people now, all of you will go and see the posters, but all other people throughout the world should know what you are doing. So 7 minutes video, including today's photo, including if you are having any setup and your affiliation and your work. Okay, thank you. Okay, so it's, please. After end of the function, all awardees are requested to come on the dais to have a group photo. We'll take a group photo of all awardees. Okay? So, yes, Jayashree, who is at the end? 1 minute only.
So, ladies and gentlemen, the Biomake in India 3 award function majestically concludes. The technical sessions will commence soon.
Okay.
After the photo session.
So, it's in?
Yeah.
Okay, thank you. All awardees are requested to come on the dais. Please. Correct. Yes. All awardees, please come here. All awardees. Yes.
A kind request, if anybody has been left behind from the awardees, to come forward.
मेल आला होता ऑटो बंद होता ना
Whether you are in the group, only I have the knowledge. So, what is your name? From? So, no, Sayar is only 1 award is there. Okay, I will be. One more. So, only 1 award is there. Take award. And take award. Please come here. I will prepare the award. Come here. Huh?
Dr. Manohar from Bangalore.
Anyone is there remaining? Sorry for mistake, but very soon we will correct it. Before giving this conference, the award will be in your hand. Okay? Only two persons are. Any more again? No.
Thank you, dignitaries. Congratulations to all the awardees and sorry for the inconveniences caused. We'll be proceeding with the next sessions.
Thank you for your attention. Please resume seats for ongoing proceedings. Thank you for your attention. Please resume seats for ongoing proceedings. Our next speaker is Mr. Chandrahas Alai, Engineering, IT and Management Consultant, Mumbai. फक्त announce करून सांगितलं खाली. त्याच्यावरती फाईलमध्ये आहे म्हणून Our next speaker is Mr. Chandrahas Halai, Engineering IT and Management Consultant, Mumbai. We privilege to introduce the speaker, Shri Chandrahas M. Halai, Engineering IT and Management Consultant, faculty at Indica Courses and acclaimed author on Indian mathematical heritage. Renowned for his books Vedic Mathematics, Inside Out and Meru Prastha, Wonder World of Indian Mathematics, Shri Halai demystifies Vedic sutras through algebraic proof for multiplication, division, squaring and cubing. A prolific writer with international journal publication, travel photographer featured in Chitralekha, former midday columnist and guest lecturer at V School, Mumbai. He bridges ancient Indian mathematics with modern engineering and computer science. Please welcome Shri Chandrahas M. Alai. I invite Dr. Wasim Bhagwan to preside over the session ahead.
I request Dr. S.D. Dekar to preside over the session, to chair the session.
Yeah. Good morning, everyone. At the onset, I would like to thank KIST, Snehal madam who invited me, Pathade sir, and I would like to kind of dedicate this lecture, my small talk, to my mathematics professor, late Dr. Sai Subramaniam, who was professor of mathematics, or the head of mathematics department at College of Engineering, Karad. So every weekend, he used to come to Satara from Karad to teach us engineering mathematics. And he is one of the major reasons why I am here. Rather, who I am is because of my maths teacher, Dr. Sai Subramaniam. So that is my Karad connection. And another connection is a long connection with Krishna Medical College, or this campus. 31 years back, almost 31 years back, my grandmother had come here for her treatment at Krishna Medical College. So it's a long connection, and I proudly say that I'm a Satarkar because I did my engineering from Satara. Yeah, so let me begin. So I think so I am the odd one out. Everyone here is kind of connected with biology, life sciences or biotechnology. I am the only one who is like, some would say, the opposite camp. So how many of you kind of relate to this image? Who is putting all the mathematics who is putting all the mathematics books in the horror section? How many of you relate with this? Yeah, students, backbenchers. Oh, one courageous girl. Anyone else who would kind of relate to this? Or rather, let me ask, who are passionate about mathematics? Yeah, anyone who is passionate about mathematics? I am the odd one out. So I accompany. Okay, so when Snehal madam called me that we are inviting you for Biomake in India, I said, remove the bio, make in India, I am coming. Are you sure you are going to kind of invite me for a bio event? She said the theme is IKS, Indian Knowledge Systems. Yeah, students, let me reveal, you can't do anything in biotechnology without mathematics. So let us look at some ancient Indian mathematics. Yeah. Yeah, fine. So yatha shikham mayuranam naganam manyo yatha tad vedak sastranam gaditam murdhina vartate. Let me translate it for you. Like the crest of a peacock, like the gem on the head of a snake, so is mathematics at the head of all knowledge. So if you want deep insight into any subject, deep insight into any subject, be it linguistic sciences, or be it AI/ML, data science, biostatistics, bioinformatics, you can't do anything without mathematics. Laukike vedike vapi tada samyike api yah. So let me just translate it. Now this thing, like why do you need mathematics and why do you do Indian mathematics? So this is a quote, or rather this thing begins with Ganit Sar Sangraha, which was composed almost 1200 years ago by a Digambar Jain Muni Mahaviracharya. And the book's name is Ganit Sar Sangraha, the gist of mathematics. In worldly life, in Vedic learning, in religious practices, in business, in everything, mathematics is useful. In romance, in economics, music, dance and drama, cooking, medicine and in architecture, in prosody, poetry, logic and grammar, and in all the arts, mathematics reigns supreme. It is used in calculating movements of the sun and other heavenly bodies. You know, it was only India who gave the calendar, the accurate calendar. So everyone now, just 3 weeks back, we celebrated the New Year. It was the Gregorian calendar. Before that, it was the Julian calendar. They were not accurate, so they had to kind of shift from Julian to Gregorian calendar. Why? Because they learnt astronomy from India and they made corrections to that. Whatever there is in all the three worlds, which is possessed by moving and non-moving beings, means animate and inanimate, all that indeed cannot exist as apart from mathematics. There is nothing in this Brahmand which is bereft of mathematics. Now, you may feel that since I am from a STEM background or other engineering and mathematics background, that is why I am like promoting mathematics. No, it is not so. Mathematics is not about numbers, equations, computations or algorithms. It is about understanding. Everyone is from this biology background, microbiology, biotech. So, who gave the laws of inheritance? Huh? Gregor Mendel. So, he was a biologist? Let me ask the question to the students. Yeah, was he a biologist? Who was he? Huh? He was actually, he used to teach physics. He used to teach physics and he was a son of a farmer. So he used to crossbreed peas. He used to crossbreed peas, matar, watana. So he used to crossbreed peas and used to record data. So from that data or statistics, he was able to see the probability distribution. And from that probability distribution, he was able to discern combinations of hidden factors. And from that combination of hidden factors, you get laws of inheritance. So if you want to know more about it, you can read my paper which is online, Mendel's piece from statistics to genetics. If it would not have been for statistics or probability, you would not have got laws of inheritance. Okay, fine. So I always say mathematics doesn't make simple things complex. Rather, it makes complicated things simple. Fine. So now, let's have some fun. Can I have a volunteer? Yeah, Shikha madam. Huh? Yeah, you can come on stage. So Shikha madam is going to volunteer and I'm going to do some exercises that mental is to do. So Shikha madam is going to choose any number from 0 to 31. And she won't tell me what the number is, but I'm going to guess it. So what I'll do is I'll go outside the auditorium, and in that 2 minutes, you can declare your lucky number from 0 to 31, including 0 and 31. Any number to the audience, I'll come in. So every one of you must be knowing the number except me. But I'll guess it. Huh?
So there's no need to go out. You can just close your eyes. By symbols, I will tell the number, sir.
सुनेगा तो सही number.
नहीं सुनेगा. Yeah.
Or you can write it and show it.
Yeah, I will do that. Just you can close your eyes, sir.
You can write it on a piece of paper and show it to them. I'll turn around.
No, they will understand. They will
Fine,
understand.
whatever.
I will choose
Okay,
a-
I'll turn around.
No need.
Wait a minute. You don't have to say anything. You don't have to say anything. Please. That is why I was going out of the auditorium. Okay, fine. Let me know when it's done.
Yes, sir. We are done.
Done? Okay. So, fine. Everyone except me knows the thing.
Yeah.
And we have no understanding. Okay, let's create a show and something like that. Okay, fine. So, I will show you some cards. You have to tell me whether the number exists in those cards or not. So, okay, let me show you the first card. Yeah. So, is your number in this card? It is there. So, I can say that it is an odd number. Okay. Now,
This number is there.
it is there. Yes, no? Okay, everyone. Yeah.
Yes. Number
Okay,
is there.
fine.
Number is there.
There.
Yes.
Okay, fine. In this one?
No, number is not there.
Okay, fine. In this one?
Yes, the number is there.
There.
Yes.
23. Anyone else would like to try it? Anyone else would like to try it? Pathade sir, would you like to try this game?
I also want to play one game with you, with the numbers only.
After the session.
Fine.
Otherwise, Snehal madam will say I've invited the wrong person. Okay, so I'll turn around. Done. Okay. I have no understanding with her. Huh? I just met her today morning. So you can't say I have some kind of, we have pre-decided the number. Okay, fine. So let's begin the game. So I'll guess her lucky number. Okay. Is your number in this?
Yes.
Huh? It is. So it's an odd number. In this one, second card?
No.
No. In this one?
Yes.
It is there. In this one?
No.
No. In this one?
No.
5? It's not. It's not in there. No. So it is 5.
Not only 5.
Huh? Pardon?
It's 2 digits.
No, but you said no, no? This one. Did you look at the cards properly?
Yes. Yes.
Okay, let's, again, I will guess it.
Okay.
Huh? Okay. The card, it is there. Okay, fine. Look at it properly.
Yes. No.
No,
No.
right? No, okay.
Only 1 digit is there, but.
Is your number is there?
Number is there.
You have to say yes or no.
Yes. Yes.
It is there. Okay, fine. So in this card?
No.
No. In this card?
No.
No, it is not there. So it is 5.
Pardon?
You s- okay,
Initially there was there.
No, no.
Yeah, in
It
this,
is- it
yeah,
is not
yeah.
changing. It is there.
It is there.
In this card?
It is there.
It is there.
There.
So now I'm changing the card.
Right.
Is it there?
No.
No. In this third
Yes,
card, it is
yes,
there.
there.
Okay, now the fourth card. Is your number there?
No.
No, okay.
No.
So it is to be 5.
Only 5, 1, 5 is there.
What? 1, 5. It is there. You said no.
What do you say?
Arre.
But I showed something else, right?
You have to look at the numbers properly and say yes or no.
No, what
You
I-
said no. And the reason why I say that you declare it to people so that you or me can't cheat. You said no.
Here, like, I don't know.
You have
what
to
to
look
say.
at the number. Okay, we'll try it with
A
someone
single
else.
number is there, but I don't.
Numbers have to be taken from 0 to 31, isn't
Ah.
it?
31.
0 to 31, and that number goes outside, out of this.
But then you said yes, no?
See, when I'm sorry, I don't know how to say. If the number is double
You
digits,
choose any other number. Look at the cards. If the number is there, you have to say yes. If
That-
the number is not in the card, you
that
have to
number
say
is with other also, I can say yes.
no. Huh?
It should be in the range of 0
Okay, okay, sorry, I made a mistake. It's my mistake, sorry. It's my mistake. I thought, okay, one more time. Okay.
Fine. Fine. Wait,
Okay, yes.
fine, wait a minute.
I'll
Wait a
tell
minute.
them.
The idea why you have to tell it to the audience so the audience knows where you are going wrong.
Okay, okay.
Wait a minute. Let's try someone else. If you are confused.
Done.
Done?
Yes, done.
Okay, fine.
Okay.
Now, is your number there?
Yes,
It
yes,
is
it's
there.
there.
So it's odd number.
Yes.
Because I only put odd numbers
Right,
here.
right, right. It was my
Now,
mistake.
is your number there?
No.
No. Audience, is she right? Yeah. Okay. The third card.
Yes, it's
It
there.
is there. Okay. In this one?
No.
No.
No. No.
It is 5.
Yes. It was my previous mistake.
पहली बार मुझे ये गेम में डर लगा. So anyways, this one. Good catch. So, now, I would like you to do some mental exercise. Yeah, I would like you to do some mental exercise. Huh? Let us say you got 2 bags full of red and blue balls. One bag of red balls, another bag of blue balls. And I give you 5 places or 5 slots where you have to arrange those red balls or blue balls. So red, blue, R, B. So you've got 5 places where you have to randomly arrange red or blue balls. So it can be all red, it can be all blue, right? It can be all red, all blue, or it can be combinations of red and blue. So in how many ways can you arrange those things? Every choice, like every place will have 2 choices, red or blue, red or blue, red or blue. So you say it is 2 raised to 5, 32 different ways you can arrange this red and blue. You can say all red to all blue. This will be 32 different arrangement. But now, if I tell you, you have to arrange them in such a fashion, all 32 possibilities in such a fashion. You have to enlist all these combinations in such a fashion. And you have to give them a row. So this all red is number 1, all blue is number 32. So if I give you a row number, say row number 13, so it will have a unique arrangement. Row number 21, it will have a unique arrangement. So number arrangement. Or if I give you arrangement, red, blue, red, blue, red. You have to give me the row number. So can you enlist all the possibilities without repeating any one of them? And in such a specific manner that I give you the row number, you give me the arrangement. I give you the arrangement, you give me the row number. So you are doing what? What is known in mathematics as combinatorial sequence generation. If you do this mental exercise or a intellectual exercise or mathematical exercise, you will realize that it's not an easy job. What seems simple? Actually, you know, in mathematics, complicated problems can be solved. But there are some very simple problems, very simple statements, which are really hard to prove or disprove. And mathematics throws at you, be it your genes. You know, you are one of the probability. Your siblings are the other probabilities of genes. Protein folding, your genes, they are nothing but arrangement of what? Proteins. Thoda bahut biology malum hai. So, can you do this exercise? I'm just giving you 5. I can give you 16 and you can arrange them. So, this thing is known as combinatorial sequence generation. Achha, now, when I'm saying for a specific arrangement, you have a specific row. Or if I give you the row number, you give me the arrangement in that row. This is what is known as in computer science or database management as indexing. So you have millions of data records, and within a fraction of a second, you have a set. Like when you send a query, the database engine gives you a set, which is the answer to your query. So who developed this mathematics? Can anyone give me an answer who developed this mathematics? And the same answer, we played this game, guessing the number game. And, okay, let me show you. My smartphone, if someone snatches it, in today's world, I'll be handicapped. If someone snatches my smartphone, or if I come here and, oh, there's no network, then what happens? I'll be handicapped in today's world. So, what is the fundamental principle on which today's digital technology works? Yes, anyone? I know you are not from computer science background, but at least you know a bit of mathematics. Yeah, anyone, any guess? It is the binary number system. And who gave binary number system? Or other, let me ask you a question. What is India's major contribution to the world of mathematics? Something even bigger than that? Yeah, anyone? No? Yes, anyone? It is the place value number system. Whether it is decimal number system or your binary number system or your base 5 octal hexadecimal or be ternary number system, that is base 3 number system. Achha, anyone into music? So yesterday we had the good cultural program, dance, music, drama, whatnot. So now no one can tell me, no sir, we are not into poetry, no sir, we are not into music. So in music, yeah, anyone into music? Come on students. If you are into music, yeah, Pathade madam. We are a great musician here. So in music you have one Yamarata Jbhana Salagam. When you are playing musical instruments, especially drums, Yamarata Jbhana Salagam. Who is a musician, drummer? Yamarata Jbhana Salagam. You have the triad, na? No, you don't have that. Okay. Anyone into Carnatic music? Maler Kota? Any poet? Okay, fine. So, who gave us the binary number system? Any guesses? Who gave the binary number system? Don't tell me Aryabhat gave 0. That's a wrong answer. We have a really stupid media in India. They do copy-paste. नकल में भी अकल नहीं लगाते वो लोग. So anyone? Great expectations from the students. Anyone? No. So it was an ancient Indian prosodist. So who is a prosodist? The one who studies prosody. And what is prosody? the science or the study of poetic meters. So it was Pingalacharya. See, it is Western world says that Leibniz gave binary number system. That is wrong. It was not Leibniz who gave binary number system. It was our own Pingalacharya 2300 years ago. He gave us the binary number system. So what did Pingalacharya do? He developed binary number system while he was studying Vedic meters. So you have the mantrochar, mantra, Gayatri mantra. You have the Mahakavyas. What are the Mahakavyas? Rama and Mahabharata. They follow specific meter or chand, or even Tulsikrut Ramayana, Ramcharitmanas. It follows a specific chand. Yeah. So, your poetic meters, they follow specific meters or chand. So he was studying poetic meters, classifying them, categorizing them, indexing them. I said indexing. You have a list of millions of names or roll calls. So election commission, it has that roll call of, you gave voting now, municipal. Just now, 6 days, a week ago, so they had that electoral rolls. So you have a chronology, alphabetic chronology, directory, dictionary. So indexing. So he was indexing poetic meters and he developed binary number system and he described it in his work Chanda Sastra, which is 2300 years old. Now binary number system, like our decimal system, is a place value number system. So in decimal number system, you have 10 digits, 0 and 1 through 9, whereas binary has only 0 and 1, yes and no. Everyone is using computers, no? Software, your data, mobile phones. Okay. So in binary number system, we have only 2 digits, 0 and 1. So you are countering only powers of 2. So when madam selected 5, Anjana madam selected 5, it is nothing but a total 1 plus 4. So I can represent numbers uniquely has just a summation of powers of 2. And then someone selected 17, rather which was that number? Shikhamar? 23. So 16, 4, 2 and 1. These are powers of 2. I can represent any integer as a summation of unique powers of 2. Hence I am not repeating any power of 2. So when you are adding decimal numbers are nothing but your a digit multiplied by power of 10. Here you are only adding up powers of 2. Okay, fine. So this is representation of 237 in decimal and representation of 237 in powers of 2, or what we call binary number system or base 2 number system. So a list of 0 to 31. And that is what I did when I was showing you the magic. Decimal to binary. So your decimal and their associated binary representation. So now you must be wondering what is the connection between poetry and mathematics. So Pingalacharya, he composed Chanda Sastra. In the 8th chapter, he has given 14 Ganit Sutra. A prosodist who is not even a mathematician, he has given amazing mathematical sutras. So the guru of all computer gurus, Donald Knuth, in his pioneering book or work, Art of Computer Programming, in 5 places he has given references to Chanda Sastra. I am a computer scientist and my books are referred in even in Princeton. So in in Art of Computer Programming, Donald Knuth says that some of the earliest and most sophisticated algorithms. So you do algo trading. You have algorithms to solve every problem, AI, ML, data science. So some of the earliest and most sophisticated algorithms for computation, combinatorial sequence generations were given by Pingalacharya and Chanda Sastra. So he developed some algorithms to enlist all the possible. So let's first understand what are Chand. What are Chand? So let's say a poetry or a mantra a mantra will have 4 parts, which are known as foot of a meter, or the pad, means 4 quarters, quarter of a meter, or you can say pad. Pad means foot. So every quarter will have fixed number of characters, or swar. Now swar, we know that Harashva and Dirgha, be it Marathi, be it Gujarati, be it Hindi, Sanskrit. So you have a short syllable, that is Lagu swar, and a long syllable or a Guru swar. Or the Haraswa and Dirgha, Choti Matra, Badi Matra. So Chand, how do you define a Chand, or how do you kind of understand a Chand? So every line will have fixed number of swar, and it is a combination of Lagu and Guru swar. So a Chand is given by the number of swar in a pad and the combination of Lagu, arrangement of Lagu and Guru. So an akshar, we say akshar, is a combination of vowel or a vowel with one or more consonant. When we say the ka means ka plus a, ka. Ti, ta plus e, ti. Or we have that compound consonant, satya, sa, half ta and ya. So you have the Lagu and Guru, Haraswa and Dirgha. So Haraswa is a, I, u, ru, l. And Dirgha, a, I, u, r, e, o, au. So we have this na tato, na mata, na bandhur, na data, na putro, na putri, na bhartyo, na bharta. Has anyone read Adi Shankaracharya? Students, I have high expectations from you. Adi Shankaracharya, so he had composed Bhavani Astakam. So this is the first stanza of Bhavani Astakam. Neither father nor mother, nor the relative nor the benefactor. So this is the second stanza of Bhavani Astakam. You alone are my refuge. You alone are my refuge, O mother, Bhavani. So this is Bhavani Astakam. You look at the Chand. Look at the arrange. There are 12 swar or 12 akshar in every pad. 12 akshar in every, or 12 swar in every pad or every line. And what is the arrangement of Lagu and Guru swar? Every quarter of Bhavani Astakam consists of 12 letters. And the arrangement is Lagagar, Lagu Guru Guru, Lagu Guru Guru, Lagu Guru Guru. And this is known as the Bhujanga Prayatam Chand. Yeah, Bhujanga Prayatam means, Bhujanga means snake, cobra, nag. And Prayatam means walking or rather moving like the snake. So it is known as the Bhujanga Prayatam Chand. So if you have 12 swar, they can be arranged in what? 2 raised to 12. That is 4096 different ways. So if you have 12 akshar, you can have around 4000 different chand. So Pingalacharya developed an algorithm to enlist all possible meters. So if you have only one swar in a chand, only one swar, so it can be either a lagu or a guru. So I'll enlist it. I'll make a table of all the possible ways. Guru and lagu. Dvikav, glav. Dvikav means pair. Glav means guru and lagu. So it's a pair of guru and lagu. So suppose now I add one more swar to the pad. So there will be 2 characters. So 2 raised to 2, 4. So what I will do is, first, I'll add a guru and lagu below guru and lagu. And then copy guru, guru, lagu, lagu. So this, the prastar, I mean say the prastar or the table or the matrix for one swar is copied inside of the second, for the table for the two swar chand. Guru Lagu, Guru Lagu, Guru Guru, Lagu Lagu. So all possible combinations are there. Now suppose if I had one more swar. So I'll copy this matrix below itself. So I'll have 8 rows and then fill the last column with 4 gurus and 4 lagu. So the original matrix for 2 is here and it is copied below, and the last row is mixed with gurus and the lagus. So you have this Pruthagla Mishraha, means you have mixed the matrix, and the Vasavastrika means the prastar for 3 swar will have 8 rows. Now I want a prastar for 4 swar matrix. So you can repeat the procedure. So it will have 16 rows. What did Pingalacharya do? I have enlisted all the possible combinations for Guru and Lagu, G and L. So if I have a row number, give me the arrangement. And if I have the arrangement, give me the row number. Okay. Now, if I replace the Guru with 0 and the Lagu with 1, I will have something like this. Look at the table. Now this table is a mirror image. See, in the beginning I had shown one table, the decimal numbers from 0 to 31 and the binary representation, that is zeros and 1. Now this particular table is a mirror image of binary number. So binary sequence generated by Pingala and the binary number that we use nowadays. So I have decimal numbers from 0 to 15 or row numbers from 1 to 16. So this is how Pingalacharya developed the binary number system, which is the most significant contribution to the field of mathematics. Not only that, anyone who is into computer programming or has done computer programming must have dealt with converting decimal numbers to binary and binary to decimal, which is known as the radix conversion. And why I was asking about that Yamarataj Bhana Salagam, drummers or musicians, they have a triad of soft and hard notes. Or when you have that cymbals, that especially Varkaris play, metallic cymbals. So they have this rhythm of triads. So these are octal number systems. It means the prastar of 3 swar. So he gave the algorithms for first thing, listing all the possible combinations to regenerate the sequence of Guru and Lagu, that is Nastam. Uddishta means given a row number, give me the sequence, or given a sequence, give me the corresponding row number. Sankhya means total number of matrical patterns. Like if I have 8 swar in a path, so I have 256 arrangements, possible arrangements. Meru Prastar, what we call Pascal's triangle. Yeah, anyone who knows Pascal's triangle? Students? Pascal's triangle. Faculty? What we call Pascal's triangle is actually Meru Prastar. When you are dealing with biostatistics, you come across probability distributions, you come across combinatorics. When you are into combinatorics, Pascal's triangle. What we popularly call Pascal's triangle is Meru Prastar, which predated Pascal's triangle by 1800, and this was given by Pingalacharya in Chanda Shastra. So he gave sutras to generate Meru Prastar. Okay. So this was his contribution to the world of mathematics. Yeah, now some biology. I hope the students are able to recognize this. Sunflower, okay. So there are many species of sunflower, and every sunflower species of sunflower will have numbers of clockwise and anti-clockwise spiral. So what is so unique about it? Anyone? Students? I thought they were like knowledgeable in biology. Yeah, faculty. Anyone? Numbers of clockwise and anti-clockwise spirals. Yeah. Harshada. You are from the town of Barwe, sir. See, it can be any species of sunflower. There are many species of sunflowers. Every species of sunflower will have unique number of clockwise spirals and anti-clockwise spirals. The number of clockwise and anti-clockwise spirals in a particular species will be consecutive numbers from Virang Sankhya, what we popularly call Fibonacci sequence, Fibonacci numbers, golden ratio. The Fibonacci sequence is actually predated by Virang Sankhya. Fibonacci himself says that I have written a book on Indian mathematics. What we call Fibonacci numbers are actually Virang numbers. So this is the relation. So if you go deep into biology or arrangement of petals, flowers, or leaves, you encounter mathematics. And Pingalacharya had briefly dealt with the Virang Sankhya, but later on prosodists and mathematicians actually worked extensively on this Virang Sankhya. Okay, so these are my books on mathematics, if someone is interested. Thank you. Any questions? Students, any questions? Yeah, anyone, any questions or queries? Okay then, I'll end my, yeah, Shikha madam. No, it means you are actually converging to that limit. If you look at, I forgot the technical name, which is the science of arrangement of petals, or what we call compound flowers, petals or seeds. In sunflower, it is the arrangement of seeds. So I forgot the name of that technical subject. So it goes deeply into math. Even shells, patterns on shells, spiral. So these are all, so basically nowadays engineers and designers are actually going deep into biomimicry. So I'll give you one more example of applied biomimicry. Paint companies, ICI or Asian Paints. So when you look at the peacock feather, you see it's a rainbow of colors. But some of the colors are not because of the pigment. It is because of diffraction of light when they pass through the micro hairs of your feather. Or even beetles. I'm not talking about the rock band, the beetles, the friend of farmers. Their color is because of diffraction, because their wings, they're micro hairs, and when the light hits that, it gets diffracted into various, when it bends, it gets, you get the spectrum of colors. So that is why applied mathematics is so very important when you are doing deep research in biology or biomimicry. Yeah. Huh? Yeah, sure. Huh? You'll have to look at that particular parameter. I'm not talking about golden ratio at the moment. I'm trying to say that they follow this pattern of Virang Sankhya. That pattern was observed. What was observed in Matra Meru, instead of fixed number of swar in a path, you have fixed number of beats. Matra, fixed number of beats or matra in a line. It can be the numbers of Lagu Guru may change, but their matras should add up to the same number. That is how we get that Virang Sankhya. And then it was observed everywhere in nature. Yeah. Thank you.
Thank you, sir. A heartfelt appreciation to Mr. Chandrahas Halai. I now request Dr. S.D. Devkar to present felicitation to the speaker. I request Dr. S.D. Devkar to preside next session ahead.
Our next speaker is Dr. Erlich Berg, PhD in Philosophy, former professor and president of German Association of Homotherapy and Agnihotra expert. Our next speaker is Dr. Erlich Berg, PhD in Philosophy, former professor and president of German Association for Homotherapy and Agnihotra expert. Dr. Erlich Berg, PhD in Philosophy, former assistant professor at Jonas University and president of the German Association of Homotherapy, leading research since 1980 across Eastern Europe and India. Collaborating with IIT Kharagpur, Tamil Nadu Agriculture University, Ferguson College, Pune. Thus, his work validates Agnihotra's effects on microorganisms, soil fertility, drug-resistant pathogens, and plant photosynthesis through modern scientific methods. Honored with Lifetime Environmental Achievements Award, British House of Commons Certificates of Excellence 2022 and consecutive Horticulture Leadership Awards. He has delivered 250 global keynotes, author of seminal publications including Bringing Home Farming into Mainstream's Indian Agriculture, Planning Commissions and the Energy Field of Agnihotra. Dr. Berg bridges Vedic fire futures with a contemporary environmental sciences. Please welcome Dr. Erlich Berg.
Okay, good morning again, everybody. So, again, I want to thank the organizers, especially Dr. Patade and Dr. Deshmukh for inviting me here for organizing this conference. Now, okay, the introduction already was done. As I said earlier, you know, we live in a time of special problems, problems which humanity has not seen before, and or maybe and and normally people talk a lot about global warming, climate change and so on. They don't talk so much about the underlying problem, which is pollution, overall pollution of the whole planet, which really brings the whole planet into danger of extinction of human race even. So, leads to a lot of problem in all areas of our life, of society. And what can be the solution? And one of the solutions which we propose is coming from the Vedic science and the Vedic science of Agnihotra and homotherapy. How can that help? Definitely we have to change our lifestyles and find sustainable ways to basically save our planet. And what is the relevance of Vedic principles for humanity as a whole? Now, in this presentation today, I will first briefly describe the method and then highlight solutions in 4 different areas: environment and agriculture, health, the corporate sector, and peaceful society. So what is the method? It's very simple. You have a copper pyramid of fixed size and shape, and in the evening I will show to everybody who's interested so you can see. You need some dried cow dung. It's important, it has to be cow dung, not mixed with any other dung. Pure cow's ghee, some unbroken, unpolished rice, akshat, some Sanskrit mantras, and the exact timetable of sunrise and sunset. Why is the timing so important? It is said that at the time of sunrise, a flood of energy comes all the way from the sun to the earth, and this flood of energy carries a subtle music. And the quintessential sound of that music is the mantra which we utter at that time. Therefore, it has to be exact mantra. If you translate, the effect is not there. And also it has to be the exact time because this flood of energy is there only for a short time. Interesting, some American physicists, astrophysicists, said the same thing. He said at sunrise, million ampere currents head in towards the earth from the sun, and at sunset they are going out again. Now things have been made very easy for us because we have an app called Agnihotra Buddy, which gives you the exact time of sunrise, sunset for every place. Now this is for my place in Germany. For here, of course, I've done the calculation today. Now, what are the mantras? The mantras are in the morning, Suryaya Swaha, Suryaya Idam Namama, Prajapataye Swaha, Prajapataye Idam Namama. And in the evening, it is Agnaye Swaha, Agnaye Idam Namama, Prajapataye Swaha, Prajapataye Idam Namama. You see, very simple mantras. 2 mantras in the morning, 2 mantras in the evening. After Swaha, you add 1 portion of rice to which you add some ghee to the fire. The whole procedure takes, say, 5 to 10 minutes. That's it. But the effects are really profound. What happens, it is said that a channel is created above the pyramid, and life energy, prana energy, which lies above the atmosphere, comes down through this channel, and an aura energy field is created around all plants nearby, and plants get nutrition, they get disease resistance, and when the flame dies, all this energy comes back and is collected in the Agnihotra ash. This is why we can use the ash for healing, we use it for plants, we use it for many applications. What's the effect on environment and farming? And Agnihotra purifies our environment from atmosphere to water resources to soil, and thus brings all nature back to harmony. And first let's look at atmosphere. You know, this experiment has been done in many, many places. You open petri dishes before Agnihotra and then send it for incubation. And after Agnihotra, you do the same thing. And you see the big difference before and after. This experiment has been done, for example, by Dr. Patade and his crew in Ferguson College, then in Desert College also. But it has been done in many, many other places also. So that is well established. And then what about, that is biological air pollution. What about chemical and physical air pollution? This has been done in North Maharashtra University in Jalgaon, but also in Ujjain. They tested with a high volume air sampler the air before Agnihotra and after, And you see that before Agnihotra, then after Agnihotra, first the values go a little bit up, but then they go down by, say, 30 to 40%. And somebody said, oh, maybe the particulates matter just settles down. So we did the same thing during the night. So we did the same thing from morning to evening. We got the same effect. One interesting thing is negative ions in air. You know, negative ions in air are an indicator for fresh air, for healthy air. If you go to a waterfall or if you go to a forest, there are many negative ions. And normally, any fire, any smoke will reduce the number of negative ions, but with Agnihotra it's opposite. We have high peaks, as you can see here. That was done in Germany, measured with a high precision instrument. And at Agnihotra time, there are high peaks of these negative ions. And that may explain why we have the effect on reduction of particulate matter, for example, also on NOx and SOx. Effects on water resources. This experiment again was done in Pune under the guidance of Dr. Patade by the PhD student whom he mentioned, Pranay Abang. He took Agnihotra ash, poured Agnihotra ash through a column of poured water, polluted water, through a column of Agnihotra ash, and you see all parameters of water are improved considerably. But not only on small scale, also on large scale it works. This is the Narmada River at a place in Maheshwar where we have one Homa farm and where I stay more or less. And they monitored the water quality upstream and downstream. And see here, the second class row, the water quality in our Homa place, Maheshwar, is much, much better than upstream and downstream. So Agnihotra also has an effect on larger water bottles. What is the effect on soil? One thing is, an ancient text says that upon this handful of soil, our survival depends. Husband it and it will grow our food, our fuel and our shelter, and surround us with beauty. Abuse it and the soil will collapse and die, taking humanity with it. And we are close to that situation. FAO, the Food and Agriculture Organization of the United Nations, says that already now 1/3 of the world's soil has been degraded compared to 1960. And they continue, if we would go on with the agriculture, chemical agriculture as we do now, in the year 2050, we will only have 1/4 of the soil per person as we had in 1960, and that definitely will not be enough. So we have to do something. We have to improve soil health. We have to improve soil health from the level of microorganisms, viruses, bacteria, fungi, algae, and so on. And in this way, create a healthy microflora and microfauna. 40 years ago, there was some test about water-soluble phosphorus in soil. Normally, you know, phosphorus is in soil, available. It's in soil, but not available for plants because it's not water-soluble. What they did is they added Agnihotra ash to soil and found out that the portion of water-soluble phosphorus increases by, say, 3-4 times. And then the question is, how come? What are the mechanisms? And one hypothesis was that there are water solubilizing bacteria. And this was tested by some experiments done in Ratnagiri, not so far from here. And they found out that nitrogen-fixing bacteria increased 100 times, phosphorus solubilizing bacteria even 1,000 times. That has been published. But also all other kinds of microbes, are affected by Agnihotter and Agnihotter ash. These experiments now were done by Dr. Ram at the ICAR, Central Institute of Tropical Horticulture in Lucknow, and they found with Agnihotter ash pathogenic fungi do not grow. On the other hand, the beneficial bacteria, they grow. So that also shows some intelligence in the Agnihotter ash. We have a problem in many parts of India with either sodic, alkaline, or acidic soil. And sodic soil, for example, there's an area near Lucknow, Kanpur, and the pH was 9.86. They made an experiment and had 3 plots, one with chemical farming, one with organic farming with vermicompost, third with organic farming, vermicompost and Agnihotter ash. And you see here, pH with chemical farming did not change. It stayed with 9.86. With organic, it came down to 9.06. But with organic and Agnihotter ash, it came down to 7.67, more than 2 points less, nearly neutral, just in one season. So it has really a big, big effect, and that could help because we have hundreds of millions of hectares worldwide with this problem. And similar with acidic soil, you know, in Poland we have a farm, the pH was 4.4, highly acidic, and agriculture engineers said nothing will grow. But with Agnihotter and Agnihotter ash, everything grew and the pH came up. You see here. Effects of Agnihotter and Homa organic farming on climate change. Now everybody talks about climate change. And as I said, climate change is just one aspect. The overpollution is the real problem. But let's look at it, climate change. At the carbon. And conventional agriculture contributes a lot to the carbon production, say 20 to 25%. And this chemical farming is a big culprit. So if you go to Homa organic farming, then the soil can absorb a lot of this carbon. Like in India, normally they say we have approximately 1%, some people say even only 0.5% of soil organic carbon. On our farm in Germany, we have 10 to 12. In some other farm, Homa farm, 15% of soil organic carbon. If this could be applied on large scale, you know, the problem of CO2 in the atmosphere could just be solved. Now, if you use that in agriculture, there are several universities which did some experiments. For example, the Agricultural University of Tamil Nadu, the station in Uti, they produced lemongrass, quantity increased, oil content increased, disease resistance was higher. Then in Dharwad, not so far away from here, Karnataka, they had a series of 4 MSc studies, and they examined tomatoes, cabbage, soybeans, and okra, and they found out yield was increased, pests and diseases could be controlled, and the quality, the nutrient content increased. That is very important, nutrient content, because that is so important for our health. You know, now we have food, okay, but if there's no nutrients in the food, you know, our body suffers. We did some interesting experiments on special aspect of Agnihotter. One is the photosynthesis activity. We, in cooperation with BHU, Varanasi, they came with an instrument to measure photosynthesis activity of plants in a Homa atmosphere and compared to plants outside. And there was a big change. And you can also see the leaves. in Homer atmosphere left and not Homer atmosphere right. So there's a big chance difference. Another research done recently, will be published very soon, is the anti-cancer efficacy of leaf extracts from plants, of Ayurvedic plants in Homer atmosphere and non-Homer atmosphere. They took curry, tulsi, neem and bale, made extracts and applied them to human cancer cells, and there was a big reduction in the viability of these cancer cells. That will be published soon. So now, one thing, if you go to organic farming, many farmers are afraid that the yield goes down and their profit will go down. And that is a big obstacle, because most of these small marginal farmers, they can't afford less profit, because anyway, you know, they are struggling. And so this was a very interesting PhD study done 2 years ago in Mysore. They analyzed 450 farms. Half of them were organic with Agnihotra, half of them were conventional with chemicals. And you see the profit per acre per season of the conventional farms. Most of them had between 16 and 20K, 20,000 rupees per season. And many also of them even had less. With the Agnihotra farms, most had more than 21,000 rupees per acre and season. And most of them, the majority has between 26,000 and 30,000 rupees per season per acre. That is a big, big difference. So that shows if people are using these Agnihotra Homer farming methods, then for them it is safe to shift from conventional farming to organic farming, and they do not risk their livelihood. They could really even do better. So that's great result of this study. So with this, we can achieve sustainable development from village level and thus guarantee global food security, which is important. Remember what FAO said, if we would continue otherwise, in the year 2050, we would only have 1/4 of the soil left per person, and that will not be enough. So now let's look at Agnihotra at human health, the effect of Agnihotra on human health. I cut this a little bit short because of the time, but for people who are interested, we have a lot of testimonies. Many diseases have been cured with Agnihotra and Agnihotra ash. Also, we have done some research. One is a project on with HIV positive children. That was done in Mysore a couple of years ago. The children were in some shelter and they were in bad state. The overall health was very bad. Then they invited me and they wanted Agnihotra, and I said, okay, but every child should perform Agnihotra himself, herself, because then they get the full effect. And within half a year, they did that, and the overall health of the children improved a lot. And then also they tested again, and the viral load also had gone down a lot. Another study was done on pathogenic bacteria. They are controlled. That was a publication done by Professor Patade and Pranay Abang. And even multi-drug resistant bacteria could be controlled. That was a study done by Professor Anjana Singh, who is sitting here. And the virulence of bacteria is reduced. Again, a study done by Dr. Patade and Pranay Abang. Now, recently, that is another important thing, the pesticide residues in our food. So we did one preliminary study in Lucknow, and we could show if you keep food overnight in water with Agnihota ash, there's a reduction in these pesticides of 70, 80, 95% even. Now this study is done on larger scale at the Agricultural University in Hisar, Haryana, because they have a residue lab and they do this systematically. This is just now going on. Hopefully next time I can report about that. This study was done in Germany recently with Agnihota ash water. They could show that free radicals are reduced, the immune system is strengthened, and cell vitality is improved just by keeping these cells in cell line in Agnihota ash water. Here you see the details. If you want, I can send you the report. Effects of Agnihota on medical potency of Ayurvedic herbs, that I already mentioned. And more such research is going on. For example, at the All India Institute of Ayurveda in Delhi, they started, I was there maybe 2 years ago, suggested they should start Agnihota, and they really started Agnihota. Since then, in the hospital, they have daily Agnihota, one room where they perform Agnihota daily, and lots of patients and doctors come and report that they really feel some effect. So it's really, really positive. And they now plan a survey on that, and also they plan a research project that just wait for the funding on the effect of Agnihota ash on growing of these Ayurvedic plants. How can, or if you think of the whole society, how can Agnihota be used in a working environment? I just want to give an example. That is from Expert Valves, a company in Belgaum, not so far away from here. Since 2012, they perform Agnihota regularly in one room, which is kept for this purpose. And also during lunch break, they perform some other additional fire, and people are happy to go there and spend their time in this relaxing atmosphere. Here's some photos. This is the factory. This is the place where they perform Agnihota. This is when many people perform Agnihota. And one, as an example, one testimony, one of the employees that says that Agnihota has significantly enhanced my ability to focus at work. My decision making has become clearer, and I'm now able to prioritize tasks more effectively. And the key benefits are improved concentration and decision making, enhanced problem solving skills, and more balanced and thoughtful responses in professional situations. That is in a factory. But of course here in a college university, you have the same situation. You know, the students, even the professors also, they have constant stress. You know, they don't know where to run first. So with this Agnihota, it gives you clarity of mind. So it helps you also in your studies. So your students up there, think of that. It could be really a good way to improve your studies and your career. Now, what is the effect of Agnihota for a peaceful society? You know, in the moment, we have a big problem of violence all over. And let's look first at the effect of Agnihota on the state of mind. One thing is pollution, a polluted atmosphere puts some pressure on our mind. It is more difficult to stay happy, to stay balanced in a polluted atmosphere. With Agnyota, you remove the pollution and um, uh, and thus your mind gets some relief. And how does it work? We have to see our mind and prana are directly connected. It is said that prana and mind are like two sides of the same coin. And the flow of prana is regulated through the atmosphere. Now, if we purify our atmosphere, the flow of prana is harmoniously, and we see it easily. This is an EEG. It was done in Moscow. This is an EEG before Agnyota, and you see brainwaves, the majority is in the beta range, 70% beta. Alpha, the more calm range, is only 17%. And after one Agnihotra, it is totally different. Beta came down to 23%, and alpha 57%. You see a big effect on your mind. And you can also see on the chakras, this is a representation of the chakras before Agnihotra. This person, you see, the chakras are very much out of balance. Just after one Agnihotra, you see nearly all the chakras are totally in line. One, the throat chakra is a little bit away, but also much better than before. So you see this one Agnihotra, which big effect that has on our subtle body. And the chakra system is important for all the functioning of all the organs. So, and the beauty of Agnihotra is this is something which everybody can easily perform at home. It doesn't need much, needs a little bit of discipline, but after you start first, the discipline develops easily. When I started, sunrise was at 4:30 in Germany at that time, and I thought it's impossible to get up at 4 o'clock. But okay, I tried. I got up, even took shower before Agnihotra, performed Agnihotra, and felt so relieved and also quite proud that I could do such a big thing. And after some time, it was a routine and never a question, oh, should I get up or not? It was automatically. So this discipline develops automatically. And if more and more people are doing it, that means going back to Vedic lifestyle, which was there in the golden age of which the scriptures talk. And at that time, there was Agnihotra in every household. And I think we should go back to that, using these ancient Vedic principles, and thus come back to a healthy, happy life of this planet Earth for all humanity. Thank you. And there's an invitation today. Evening Agnihotra is at, you see here, 18 minutes past 6. We have to see where we can perform. I don't know what the schedule of the conference is, but hopefully we can do it either today or tomorrow so everybody can experience. 18 minutes past 6. Say 6 o'clock we should start. Okay. Okay, we'll see. Dr. Patrade will announce it. Okay, that's it. Thank you.
दोनला सांग म्हणजे 2:15 पर्यंत येते.
Sir, the session is open for question and answer. Thank you, sir.
Thank you, sir. The session is open for question and answers.
Hello. Hello, sir. Thank you for presenting multiple applications of Agnihotra in various disciplines like microbial treatment, environmental treatment, or healthcare, like treating cancer, or various application for Agnihotra. But I am curious, like how does this Agnihotra acts on, like if we are treating microbes? So how it works, where it can work, and how these microbes are killed, or what is the mode of action or mechanism of action, how it works, and how that Agnihotra kills microbes. Or if we wanted to treat cancer cells, so how it act on specific, does it act on specific targets, or randomly it kills cancer cells?
Okay, very good question. One thing is, Agnihotra ash does not kill anything. It changes the environment, so these cells, these microbes, harmful microbes, do not develop. It is an ahimsa method. Ahimsa is an important principle of the Vedas. But, as I said, it's a very good question. And see, we are only at the beginning of this research. What we have found and what has been confirmed in several studies, like the bacteria in the air, reduction of these bacteria in the air, that has been repeated a number of times. So the effect is clear, that's established. But how does it work? We do not know. One of the hypotheses is that the negative ions in the air have an effect. That is a possibility. But there may be other mechanisms also we do not know. So here are a lot of young scientists, so that is still a great chance for them to work on that. There are many, many topics for very good MSc thesis or even PhD thesis in that area.
But what is your suggestion, how it can be validated, like if people want to work on that? So how it can be validated by using multiple approaches, like which approaches can be used for validation?
Oh, see, I'm not an expert in microbiology, so I do not know that. But the first step would be to repeat the experiments which were done earlier, so that the scientists or students are convince themselves that the effect is there, and then they have to find out what are the possibilities. I mean, if you think of negative ions, you could work in this measure negative ions and see what's the effect on bacteria. Then, I mean, one thing is the virulence of bacteria is reduced. That has been done by Dr. Patade and his team. So that is already one thing which would explain that some health benefits. Yeah, because if the virulence of bacteria is down, then you don't have this big problem of these bacteria. But see, for that you would need an expert in microbiology. Yeah, there are many, so you can discuss with them. I would also be interested to hear about these possibilities, but I can't really give some valid suggestions.
Then there is a great scope for research. If it is really working, so there is a great scope.
Yes, definitely so. And very interesting. It is not just repeating other research, it's very interesting new research, and it's very important. You can help humanity with it.
Any other question from audience?
Actually, I was thinking about this Agnihotra and the Havan which we used to do at our homes, and to answer the question actually how it kills the bacteria or the microbes. I, first of all, I am not expert and I actually have no scientific validation, but as a curious person who used to do these Havanas at home, we hear that the original ingredients in Havan were also as a Havan Samagri. And in ancient times, since Havan Samagri contains the lichens, specific lichens, and it was thought and believed that if you burn these lichens, it increases the oxygen concentration locally. And because this oxygen concentration increased locally, it has some antimicrobial activity, so that it purifies the local environment where you are performing this thing actually. Nowadays, this Havan Samagri, if you go and buy in the market, I don't know what it is made up of, but initially it was basically a specific lichens which were originally carried out from mountainous regions or something like that. So, and the other ingredients also include in that. Not especially for Agnihotra. And my question, sir, these Havans we used to perform at different intervals at our homes. So, do you suggest that these to be performed in the morning and evening on daily basis to have a good result, or?
I know that in India there are a lot of Havans, homas, whatever, and I do not know much about the effects. With Agnihotra, we know because we have done a lot of research, and for Agnihotra, we do not use Havan Samagri. We just use cow dung and ghee and some rice. And about the hypothesis with the oxygen, I have heard that, but it is always only claimed, and I've not seen any study where they actually measure the oxygen concentration. I mean, if there would be an increase in oxygen, that would be great, but I doubt it because, you know, just if you look in the chemistry, you burn some substances, and substances contain carbon, yeah, cow dung, ghee also, and you burn that, and what is the effect? You will have CO2. That means less oxygen because the oxygen comes from the air. So I doubt that actually by doing these homas in the environment itself, there will be more oxygen. If you look on larger scale, definitely yes. What we have seen, the effect on plants, photosynthesis activity is increased, so definitely there will be more carbon. But that is, if you perform it here in the room, that will not happen. It will happen if you do it in nature, then yes. Otherwise, the immediate effect, I would not.
Yeah. I also don't know, just as curious, for the curiosity, I had heard somewhere that it releases nascent oxygen ozons also. So, but I'm not sure.
Okay.
Yeah.
Okay, any other question?
Sir, only the last question. We microbiologists, we had done one experiment that how is the growth pattern of microorganisms in the new moon day and the full moon day. So we observed that in the new moon, the growth of the microorganism is more, even we get the new leaves comes from the plants also. So the growth starts from the new moon. So have you observed anything, any changes during this full moon and the new moon because of the Agnihotra, any reduction that, or every day you mean the microbial flora is reduced?
There is a lot of research done in biodynamic farming regarding these full moon, no moon cycles. Even other planets are also considered.
Even on eclipses as well.
That has an effect. And what we know is that during full moon and no moon, for example, there's a higher incidence of accidents. Hospitals know that. they're prepared for full moon. They know there will be more accidents, more patients come. So what we do, of course, Agnihotra we perform every day, but what we do in our Homa centers on full moon day, no moon day, we increase the number of hours of the additional fires which we do.
Yeah, that is what, because it is non-fact. So that is I wanted to know like how that affects. So
okay,
thank you, sir.
Thank you. Okay, nobody else? We can go for lunch?
First of all, it's a great information. I did not thought regarding like Homa therapies like this. I did not heard about this before, but it is very interesting topic. Today's we can see population is increasing day by day. How I want to ask that how much it will reduce help to pollution. Pollution, sir.
See, every single Agnihotra which you perform reduces pollution of the air in the room where you are, of the plants nearby, of water and the soil. So the idea is if more and more people are performing Agnihotra, then the range will be bigger, and in the end the idea is if Agnihotra will be performed all over the globe, then we will have a global reduction in this pollution.
Okay, sir, thank you.
Hello. I think at the time of lunch you can interact with the person regarding that, and we will close the session now. So, okay.
Sir, I request our session chair to felicitate our resource person. Thank you, sir. And I request Dr. Dhanushri sir from KIST to felicitate Devko sir for chairing the session. Thank you, ma'am. We are breaking for the lunch and we are requesting the participants to reconvene at 2 o'clock for the next Hello. Hello. Good afternoon, everyone. Good afternoon, everyone. Welcome back to the afternoon session. Let us now begin the next part of our program.
Hello.
Hello. Good afternoon, everyone.
this linear model is creating. So we are developing the technology in a way that you take from the nature, make it, but dispose it in a way that it goes back to the nature. And when it goes back to nature, it doesn't create any harm to the nature. And when you are making it, at the same time, you should not create any harm to the nature, right? So while doing that, what we receive is, when I'm talking about the feedstock today, I'm mostly talking about the tannery wastewater. So the wastewater you receive from the industries, they are highly loaded with lots of nutrients, and those nutrients which we look as a waste is actually not waste. That can be further exploited to be used as a resource. And that's why the whole project fits very well into the circular bioeconomy model. And while in order to achieve this target, microbiology and biotechnology is the key factor, because the microbes are the one which are closing this linear loop and making it a circular loop, and then is contributing to the circular bioeconomy finally. Now, we talk a lot about the India's traditional ecological wisdom. Now we have more experts who have more keen knowledge over this. But rather than going into something very complex, my suggestion is, if you work at home and if you are observing what you are doing or what your mom is doing, every part of the lifestyle, especially the Indian lifestyle, has a microbiological input in it. Go to your kitchen, help your mama, and you will see when she is making a dough, she is allowing it to rest for some time. You will see when she is making a curd, how a small rennet is changing into a whole bowl of curd. You will see when she is making a batter for idli, how she allows it to ferment for a day or overnight, and suddenly she can make a idli which is so fluffy and very nice to eat. So you have been enjoying to eat, but this is the time to just watch mindfully what is happening at home, and how your father is maybe helping in the garden, and how he is composting the stuff, how he is using your kitchen stuff and making, the land more fertile in your garden. So every part which is a part of our Indian lifestyle is basically our heritage knowledge, which we are actually taking from generation to generation. And these knowledges are now getting validated scientifically through the metabolomics, proteomics, microbiological assays. So there is nothing basically new in it. It's just we are validating it. So if we keep ourselves rooted and we go back to our ancestral knowledge, we have lots of motivation already happening there. Okay? So our traditional ecology and is actually finally converging to the modern science which we actually acclaim nowadays. Coming back to the feedstock, which is tannery wastewater, it is a huge and massive problem all over the globe, not only in India, because nearly 80% of sewage remains untreated, and only 38% of industrial wastewater get treated properly. And believe me, these are the data which we are receiving for 22 countries, but among those 22 countries, the developing countries are generally having the most problem. So the idea is to remediate this discharge in a way that the pollution level of discharge come to a nominal level, and you can discharge it very safely into the nature, whether it is going to the landfill or it is going to some water bodies. So if anything is getting discharged back to the nature, the pollution level has to come down to a level that it remains not at all toxic to the environment. Because these discharges are not only very high in organic and inorganic content, but at the same time, they contain certain ions which are of very toxic level, like they have high levels of lead, sometimes chromium, that if are discharged without any further prior treatment, then they will actually create problems for the aquatic and the land life. And generally, just for the sake of students, I like to state that whatever project you are handling, if you are looking for a future fellowship or if you are looking for some future projects for yourself, it's always good to correlate your project, your plant project with the United Nations Sustainable Development Goal, because if it fits to any of it, you will have a priority, and there will be higher chances of you getting your project getting selected. So, like for the wastewater, it fits completely with the SDG 6. But it is not only the SDG 6, even SDG 9 and other sustainable development goal things also fits well into it. So, like the alternate energy, because when you are valorizing it, you are producing certain byproducts like ethanol, butanol, or even bioplastics. So, the suggestion is, whenever you are planning a project, plan in a way that globally it is basically accepted, and if you can link it to the United Nations SDG goals, then that will be very helpful for you. So I have been using this term a lot, which is bioremediation. So what is bioremediation? Bioremediation is very simple thing. If I talk in a very layman language, you use certain biological resources like microbes, you can use plants, or the enzymes extracted from plants and microbes, and then you use them to treat the waste or wastewaters. And while doing so, you reduce the pollutant level of that waste or wastewater. So this can be achieved in two ways, which is in situ and ex situ. In situ, in a broader language, if we say in situ is the process which you perform or the treatment you perform on site. So if you are doing the treatment on site, on the field only, say via biostimulation, augmentation, like directly putting microbes there, or directly adding some nutrients to the soil, then it will come into the in situ. While ex situ is the one when you take a sample from the site and you take it to the lab, and then you do work on it. You use bioreactors or the flask or the other technologies to remediate the whole the target waste. Then we say it is ex situ. So today what I am presenting is all lab work. So that is the ex situ work. Now, if you compare in situ and ex situ, generally ex situ is more expensive, and in situ is less expensive, but in situ is generally a little non-specific, and it is also very slow process, while ex situ is a very specific, and it goes faster. So when you are doing some experimental optimization, and when you have some deadlines to meet, you will always prefer to have the lab scale experiments, and then you go for the ex situ. Tannery wastewater, I don't know how many of you know what is tannery. Tannery is the industry where they take animal hides, and they change into the leather, and while doing that, they do lots of processing. Like the leather undergoes soaking, liming, deliming, pickling, chrome tanning processes. It undergoes all these processes, and while doing that, lots of harsh chemicals are used. So at the end, what happens, the water that comes out of the effluent that comes out of the tannery industry is having high salinity, alkalinity, organic and inorganic loads, and suspended colloids, and even those fats and proteins, the animal protein that comes from the skin, all the leftovers, even the hairs, everything is there, right? So you cannot dispose this highly loaded wastewater into the nature directly. You have to treat it, right? So what you do is, there are generally three ways of treatment. The primary treatment, in which it helps in the industry itself. What they do is they screen or remove the larger particles, like the grit removal, and they do the neutralization because to balance the pH. And then they also try to add some chemicals to do coagulation and flocculation, or allow it to settle in a settling pond, which is a huge pond there, and it allowed there for a month or 2 or even 6 months, so that all the heavy things get settled, and then they take out the upper part and then they process it for the secondary pre-treatment. Secondary pre-treatment is not something which is essentially available in all the industries. It depends upon what are the policies of the country or the region. But wherever it is available, like in South Africa, Cape Town, we do have some batch reactors, like which are anaerobic digesters, or even the sequential batch reactors, and some lagoons which are there in the industries to treat the primary treated tannery wastewater further, and so that the chemical content of the pollutant is further reduced. Finally comes the tertiary treatment. My work is basically on the tertiary treatment, what I am presenting today, and I'm using microalgal bacterial consortium for it. Consortium is nothing, it is the co-culture where I use certain microalgal cells, and I use some bacteria. They live together in harmony, and together, when they are living and growing, they remediate the wastewater. So coming back to tannery wastewater, the most challenging part is every batch of wastewater you receive has a very different composition. Like you can see, there is batch 1 and batch 2. If you can check, then you see that there is a vast difference between batch and batch 2, but they come from the same tannery wastewater industry. So sometimes what happens, the secondary unit fails, and the effluent which is released from the industry, it gives a very strongly loaded wastewater, and sometimes it works efficiently. There could be possibly many reasons, but this is for sure that every batch has its own composition. So the major challenge is growing a consortium which is very effective and is effective for all the kind of batches which it is receiving. So as I've said, the microalgal bacterial consortium is like growing 2 to 3 types of microalga together, and then allowing it to grow in wastewater, tannery wastewater, and then while doing so, allowing even the bacteria to grow along with it. And this whole process is like optimization, which takes time, and once it is achieved, then you have an inoculum which has right amount of microalgal biomass along with the right bacteria that can together remediate the tannery wastewater. I will share the process of optimization in very brief in my upcoming slides also. So the interesting part is that they live in harmony. Why? For the reason that we all know microalga is photosynthetic. So while doing the photosynthesis, what it will do? It will take the carbon dioxide and will produce oxygen. While we know bacteria, it needs oxygen, and it will give carbon dioxide. So what algae is producing, bacteria needs it, and what bacteria is releasing, algae needs it. So they are very happy with each other, right? The only thing for which they fight is the carbon source, and that carbon source is the wastewater because its organic load is very high. They both need nutrients to grow, and the basic need is organic load and the nitrogen for sustaining life, right? And these both things are surplusly available in the tannery wastewater. It is available to the extent that sometimes we need to reduce it because it is so high it doesn't allow them to grow there. So these are the various metabolic pathways it undergoes. Like when you grow these consortium in the tannery wastewater, certain chemicals get bioaccumulated inside the cells, the microbial cells, and certain gets transformed. Certain mineralization also happens, while certain gets biodegraded, and it definitely, because they are the living system, they undergo aerobic respiration, and as I have already stated that the microalgae will undergo photosynthesis, and there will be a conversion of sulfide, and the bacteria are generally nitrifiers and re-nitrifiers, so they fix ammonia and convert it into nitrite, further into nitrate, and then they release this nitrogen as a nitrogen gas. So it's a very interesting but very complex reaction which happens there, and but at the end of the whole thing, your water is remediated. So if we go for the systematic of microalgal, bacterial, tannery wastewater biorefinery, this is the basic biorefinery. So this influent is basically coming from the industry. We have this high liquid flow channel reactors made by one of my colleagues. This flow channel reactor has the capability of removing sulfur from the tannery wastewater. Once the sulfur is removed, because sulfur is very toxic for the living organisms, then once the sulfur is removed, we take it to for the secondary pretreatment, which is through the anaerobic digester, and then further the biogas is released, and rest of the effluent, it goes to our photobioreactor where the MAB is growing, and then at the end of the whole cycle, what you receive is a clean wastewater and the biomass, the algal biomass, which can further be processed to produce some value-added products like biodiesel or animal feeds, or even the base for the PHA production. So how this work starts? This is a very basic experimental setup which I have shown you graphically to have some idea. So what we do, you take the wastewater from the industry. Now this is so highly loaded that it took nearly 8 months to optimize and to find a right microalga that can sustain the high concentration of organic and inorganic load which is there in the tannery wastewater. So we started with using just 10% of tannery wastewater, try to grow our inoculum there. Then and then we screen, like you start with 10 to 15 cultures. Among those 10 to 15 cultures, only 10 are capable of growing, and then you screen further and steadily you start increasing the concentration of tannery wastewater 10%, 20%, 50%, and at the end you will find 2 to 3 strains or maybe 4 or 5 strains which are now strong enough, which are very resilient and can grow in 100% of tannery wastewater. This process looks easy, but this is very time consuming and this is very depressing sometimes. But at the end, you will find your results and you will find what is the right alga that will work for you. So once you have the right alga with you, you know these are the cultures which can sustain in high concentration of tannery wastewater. Then you do the molecular identification of it, right? And then you identify using them some specific algal primers. And once they are identified, then you do the microscopic confirmation using confocal microscopy as well as the SEM and TEM. And once this is done, then you know you have a completely profiled inoculum with you. So, like for my work, I have made a consortium with 4 of these cultures, which is Tetradesmus obliquus, Tetradesmus dimorphus, and another strain of Tetradesmus obliquus, and the Neochloris. And you can see they all are growing sufficiently in tannery wastewater, but when you make the consortium, they are able to grow individually as well as a consortium. So once this is sorted, you plan the flask scale study, and you need to know that whether this process is feasible for all seasons, because you don't want to keep whole thing in your lab. It needs to go to the field. So once it is in the field, it will be exposed to the seasons, all the seasons there. So what I did was I checked the effects of all the seasons on these consortium growing in tannery wastewater, and then I also analyzed the biomass, what is the content in that biomass, and how can I valorize it further. Once this has been standardized, then I upscaled my process, and from flask scale I came to the bioreactor scale, and here you can see I have a bioreactor, a set of 2, because we generally do all the experiments at least in replicates. So it goes with the 20, which it is having a capacity, a working volume of 20 liter. So we perform the same optimized process here, and then we try to analyze what is the growth profile in the photobioreactor, how you can compare it with this, and what is the seasonal effect, and how, and finally the biomass what you are receiving, what is the composition of that. So this is the photobioreactor I'm using in the lab, and its annotated diagram. So for the seasonal, optimizing the lab, optimizing everything in the lab condition is quite easy, because you have reactor which have all the where you can easily maintain the temperature, the RPM and everything, and we mimic it with the average climatic data we have received for the Cape Town, which we have received, and from the South African Weather Services, and we just mimic the same data to our flask scale experimental optimization. So you can see, as it is very well expected, that the tannery wastewater will have a very high COD, and while doing that, and there is 2 types of CODs. One is the total COD, another one is the soluble COD. The easiest way is to understand is you just remove all the suspended particles there by centrifuging it. Whatever supernatant is left over, you take a COD of it, then you will say this is the soluble COD, and whatever was in the total, that will be the total COD. In that way, you also come to know that how much COD is contributed by the biomass itself, and how much is contributed by the water itself. So at the end of our main target component for remediation was the ammonia. The initial ammonia concentration was very high, which was above 40 to 50 mg per liter, and the target was to bring it to below sustainable, below allowed limit. And at the end of it, we have received it every time very efficiently. So this is the remediation potential under stimulated climatic condition, definitely under the flask studies. So you can see that ammonia is reducing, and the total alkalinity and the pH is also coming to the normalized level, and the measurement, and this is all about the microalgal monocultures, and the dark line what you are seeing is basically the consortium. So you can see a consortium is behaving more nicely than their individual microalgal culture growth. Now, if we compare the total COD and the biomass measurement, you will see that the most effective season for the bioremediation was observed to be the summer season, and because the microalgae grow very effectively during that season. And winter, it has been a little challenging, but the good part is for all the seasons there could be a difference in the remediation, but it allows remediation in all the season, and it can sustain itself in the extreme weathers also. So even if the remediation scale is a little fell down, the inoculum is going to stay there and it will start processing. Sometimes it's just if summer is achieving it, say in 15 days or 20 days, winter may take 40 days. And at the end of this, whatever biomass we receive, we characterize it, because we need to know what is the composition of that. We are more interested in having the protein, carbohydrate and fatty acid profile along with the other elemental profiles, because as you can see here, the protein is comparatively higher than the carbohydrate as well as the fatty acid for this set of inoculum, right? For this set of biomass. So what we do is, according to what we have received, we say because the protein content is high and even the NPK is good, this can be the biomass which we have received can be further exploited as an animal feed or even the fertilizer or biofertilizer or the biosupplement. But when we work with some other wastewater and with some other set of this microbial consortium, we get a very different profile. And maybe in that profile we have higher carbohydrate content or higher fatty acid content, and if the fatty acid content is high and it has a good amount of fame, then we can say that we can further extract the lipid from the biomass, transestrify it, and you can attempt to produce biodiesel or bio-oil from it. So this is a very promising technology where you can vary the tannery wastewater, and on the basis of that you can develop a new consortium, and that consortium can give you a specific desired results. Now if we come to limitation and constraints, as I have already discussed, the very high salinity and the hardness of the wastewater is the most challenging part, because only halophiles can grow in that high concentration of salts. But the microalgae are very magical. They tend to adjust very nicely. So if you allow them, if you give them some time, maybe few months, you will find that what was not growing initially, after few months, they become very they started showing very promising growth, and because of their resilience, they suddenly develop a very good biomass. Toxic metals in the wastewater is the major challenge. Previously, when the tanneries were processing the hide, there was a process which is known as chrome tanning, and for that they use chromium, and chromium is very toxic. So that was very challenging, but thankfully the policies has changed, and the government has now banned the use of chromium. So instead of that, now they are using another technique, which is use of vegetable oil tanning, which is now very eco-friendly and doesn't need any prior pre-treatment. So it's very helpful. Definitely, as I have said, the variable wastewater composition and the competition of food between algae and bacteria, and sometimes the difficulty in harvesting the biomass, because every biomass has its own tendency. Like the one with which I am working, they tend to coagulate. We call them synovia. So they tend to coagulate after certain days, make a ball of kind of thing, small little balls, and they settle down at the bottom. So for this consortia, it was very easy to harvest the biomass, because biomass was already settled at the bottom after say 3 to 4 weeks. So it was sorted. But not all the algae tend to do the same thing. Sometimes you need to undergo certain process like frothing or so that you can just collect them, and so depending upon the culture which you are using, you can select the harvesting process for that. Scaling up is always challenging because what happens at flower scale, it's very different when you bring it to a reactor scale. And further, from the lab, when you take your reactor to the field, it behaves very differently. But believe me, it is just a matter of time. You give some time to these magical consortiums, the microalgae and bacteria, and you will understand that they acclimatize quite fast, and after a certain time you will feel that okay, now they have started giving a very positive result. Because when I started these experiments, every experiment was running for 60 days, and today within 2 weeks I received results. So this is how they improve themselves or acclimate themselves, and that's why I have in the morning I've also said that the persistence and patience is the key to research. So you have to be very patient when you are researching. So future direction, definitely we want to develop more stress tolerant MAB consortia for different kind of wastewaters. We can always improve the reactor design in a way that it is more economic and at the same time it is more productive. We are applying now proteomics and metabolomics to understand the biochemistry behind it, and try to find better ways for the harvesting. And we do all the designs through statistical optimization using various softwares like Minitab, Statistica, and design experts, which help us in designing a model for the experiment and give us that what are the factors you want to work with, and what are the levels of different factors you want to work with, and what kind of study do you want to do, whether you want to study through CCD or RSM analysis. So it's very much easy when it comes to using software. They sound big, but believe me, once you start using it and you get friendly with it, they are very easy. So the future is to generate more value-added products. Like currently, we are focusing more onto the high-protein products and using them as a biofertilizer and animal feed. But we are looking forward to develop another inoculum in which maybe we can develop an inoculum which has higher lipid content or can also give higher carbohydrate content, because one of the parallel projects is going on which we are attempting to produce bioplastics through microbial route. So if we can generate good amount of carbohydrate through this, then this can be further used as a feedstock for growing some actinobacteria, and that can convert these into PHA and we can make bioplastic of it. So I'd like to acknowledge my team at MB, who is always very supportive and have been very nice in completing this project, and our collaborators which are from University of Cape Town, our funding bodies which are Water Research Commission, and definitely a big thanks to the organizers of Biomake of India 3, Microbiologist Society, and Krishna Institute of Science and Technology, Karad. Thank you so much for hearing me. And these are my publications, recent one, last year ones mostly. So if you want to go, you can definitely check them. The one thing which I want to say is, as it's written, that the nature never works in isolation. So we need to know that the nature never works in isolation, so we also cannot work in isolation. Always work in team, be a consortia of your team, and be a productive one. Good luck to all. Thank you. Any questions?
Now the session is open for question and answers. Any questions?
Yes, sir.
Excellent presentation.
Thank you.
I come from a medical background and work at a medical college as a professor.
Medical, okay.
Professor of microbiology. What was interesting was the wastewater treatment, which you touched. And of late, we keep teaching to students that what's supposed to stay in the hospital is not staying in the hospital. It is seen in the communities.
Yeah.
So all the drug resistance bacteria, which we mainly, concerned, they are in the community now. Not just the community, even it is seen in the soil.
Yeah.
Whatever we grow also, they have a drug resistance bacteria.
Yeah.
And we tried a different mechanism. We enhanced the chlorine to look into it, but still, the bacteria are resistant to chlorine. And as you rightly said, the bromide, it is not used. And ozone, it's not working. And in your study, have you come across any drug resistance bacteria after the treatment, first point? And we developed a technology to overcome it. We called it as a biodisinfectant.
Yes.
Right? You just add a bacteriophage, months together, it takes care of all notorious, pan drug resistance bacteria, so that it doesn't enters into the community, doesn't enters into the municipal facilities, nor into the drinking water. Right? So, what's your experience on this?
To be very frank, what I'm presenting is in South African context. And there the policies for disposal of the medical waste, the industrial waste, the municipal waste, it's actually very strict. They don't allow you to dispose anything like that. So when we were working with lots of wastewater, we only came across the microbes which belongs to that community. I mean, I didn't find the drug resistant or those. And to be we haven't worked for a very specific group of, microbiota. It is whatever we have done is for a general one. And you will be surprised to hear, but all the process what we have done at flasks, especially at the reactor scale, it is not at all sterilized. This is whole unsterilized procedure, because when you take it to the field, you cannot work in sterilized condition. So it is on-field application needs something that can be done. And because of the strong policies of government, we didn't came across something which is, and this is more about reducing the chemical pollutant rather than, working on the specific or targeting certain specific microbes or reducing them. So my experience in that area is actually not very relevant, because we didn't find anything, probably because of the government policies. That is something which we need to take out, which concerns us a lot.
Because we tried, in the ETP till 6 ppm of chlorine.
Yeah, chlorine is
But with 6 ppm also.
Yeah.
There were good number of hospital flora survived.
We always but prefer not to use chemicals, but I know sometimes you don't have options, right? And
that's a normal disinfectant procedure in the STPs.
Yeah, that's a
and
regular
ETPs.
one which happens everywhere, that I know. But
Thank you.
I think I'm not the right one to comment there.
Thank you. Thank you.
Any other questions?
There is one.
Hello, madam. Nice
Hello.
presentation.
Thank you.
So I have a few questions to you. You are using microalgae and bacterial consortia, okay? So those bacteria you are isolated from anywhere, or those are phycospheric?
No. not phycosphere. These, as I said, I'm growing them in unsterilized condition, right? So when I was developing my inoculum, I was developing it with tannery wastewater, correct? So when there is two processes which are natively associated with this. When it comes to bacteria, it is nitrification and denitrification, because our target component is to reduce the ammonia concentration in it, correct? So when you develop the inoculum, and what I did, I added specific amount of microalga culture, known cultures, to my tannery wastewater. I allowed it. So it's the natural denitrifiers and nitrifiers that after a while they start growing in it and they start remediating the wastewater. So you don't need to add them or supplement with bacteria. It's just the microalga which has been added in a specific count on the specific number or concentration. The bacteria, when you develop your inoculum in unsterilized condition, they grow naturally there and they start doing their because this is a part of natural cycle. I'm not synthesizing anything, or I'm not modulating any bacteria or its gene to start doing some process. We are focused more into the natural process. So this is a natural way in which the natural denitrifiers and nitrifiers start growing after some time to remediate that feedstock.
It means that the symbiotic microflora of microalgae growing. So are you claiming that after wastewater treatment, there is no any toxic element in that water?
Yes, we go for complete ICP analysis and CHNS analysis to know the elemental and complete profile of that wastewater, to know what element or what is under the limit and what is not under the limit. And to be very frank, when it comes to when I'm using tannery wastewater as a feedstock, I cannot go for valorization that can suit the human supplementation, right? Because it has certain amount of chemicals which neither microalga take nor bacteria takes. So especially with the tannery wastewater, we cannot use it for human supplementation, even if it is producing high amount of protein. Otherwise, if I'll be using some, say, maize mill industry effluent, right? Then I can use it for that purpose.
Okay.
But we always do complete profiling of the final remediated wastewater to understand and to decide the valorization way of it.
I think tannery wastewater may contains the dyes in it, or
it always contains dyes.
So after dye degradation in the tannery wastewater, there are maybe some chances the byproducts generated are maybe toxic or not.
Dyes, for some reasons, the dyes are removed at the primary level only, what is done at the industries. Because the one which we receive, it only contains some blood and some skins, some those kind of proteins, but it doesn't contain dye. Because as I've said, the primary and secondary treatment, they try their best to clear lots of unwanted things. What they can't clear, they send it to us so that we can give them a solution for that.
So it also contains the synthetic organic material in your tannery wastewater, or no?
What do you mean by synthetic organic material?
Synthetic organic material in the sense, just like a dyes which are using during the tanning process, or?
I don't find any specific dye there.
Okay.
Yeah. What I'm observing is the hides which are there, which are processed there, are mostly ostrich and even the bovine, but generally they never contain dye in our supplements.
Okay. Thank you so much.
Thank you.
Any other questions? Okay, thank you so much everyone. Warm gratitude to Dr. Amrita Ranjan. I request Dr. Aparna Pathade to felicitate the speaker. I request Dr. Dhanashri Patel to felicitate the chairperson. Thank you so much, everyone. Next, we have Dr. Paramananda Burman, scientist and head of the studies in Science Communication Division at CSIR, National Institute of Science Communication and Policy Research, New Delhi. He holds a PhD from the University of Delhi in botany and conducted postdoctoral research at ICAR NBPGR, specializing in plant molecular biology and bioinformatics. As a co-PI for the Swastik National Initiative, he manages the dissemination of scientifically validated traditional Indian knowledge across the digital and regional language platforms. His professional responsibilities include serving as associate editor for the Indian Journal of Traditional Knowledge and the Indian Science Abstracts. A recipient of the 2019 Indian Science Media Fellowship, Dr. Burman has authored numerous research articles and over 150 popular science stories to bridge the gap between complex research and public understanding. Now, I request Dr. Vishwas Patil, Associate Professor and Head of the Department of Microbiology, Lal Bahadur Shastri College, Satara, to chair the session ahead. Over to you, sir.
Thank you. Respected Chair, dignitaries, and the students and participants of this conference. At first, I'd like to thank Dr. Pathare for inviting me and giving this platform for interaction. As the last speaker mentioned, it's not easy to interact in a post-lunch session because all of us are a bit sleepy. And then generally attending lectures, back-to-back lectures is also very difficult. That's my personal experience. So as I have traveled all the way from Delhi, so I'd like to give you some take-home message, at least I'll try for that. So I am from CSIR NISPER. How many of you know CSIR? I think all of you know. At one point, you will appear at least the exams and all. And then, so we have 37 labs across India. And my lab is National Institute of Science Communication and Policy Research. So we take science to people. So what happens in research is we do research in lab and then we publish research papers. Research papers are in highly technical language. And then common people cannot understand those research papers. But to build a progressive society and to ensure development, we must take science to everybody in the country. So we are trying to bridge that gap. And then we are working in this domain called science communication. By the way, how many of you know that there is a branch called science communication? Any student? Okay, very few, no? Okay, good. So as I mentioned, that science is coded in very technical language, people cannot understand. We try to simplify those information and take it to people. So first take-home message is that today you have learned about a new branch called science communication, where you simplify science. It may be a story, it may be a video, podcast, through social media, these days reels, and then in various languages. So in different ways, you take it to people. So I'll come more into science communication later, but for the students, you can also make a career in science communication like I have made. So I was a plant molecular biologist, but then I shifted completely towards science communication. This is our campus. So we have two campuses in Delhi. One is Pusa campus, one in Satsang Bihar campus. By the way, we publish 15 research journals also. So in different domains of science, you can communicate your work. Today, I'm here to talk about traditional knowledge. So this session is mostly on life science and traditional technologies, but I'll do an overall glimpse of Indian traditional knowledge with scientific basis. I'll definitely touch life science at some point, but it is not entirely on biology or life science. So also, when we talk about traditional knowledge, people mostly think it's only about Ayurveda or traditional medicine. But it's not like that. India is very rich in multiple domains of science like astronomy, mathematics, chemistry, physics, agriculture, metallurgy, architecture. Take any branch of science, we have very rich traditional knowledge. And these are more than 5,000 years old. So there are multiple efforts now to preserve our traditional knowledge, do more research, and then disseminate. So some of these are like the R&D organizations, the my organization CSIR, then ICR, Ayush, all of them, they are trying. Other higher educational institutions, NGOs, and at grassroots level also, many people are working to promote traditional knowledge in India. So what my institute is doing, many of you might have heard about the turmeric, revoking of turmeric patent by India. So they gave a patent on use of turmeric. Then we filed a complaint, and then eventually that patent was revoked. So it was done by my institute. So now it's a different branch of CSIR called TKDL, but TKDL was originated in the organization I'm working in. Then we are running a journal called Indian Journal of Traditional Knowledge, where we publish research work in different domains of traditional knowledge. We also publish one encyclopedia called The Wealth of India, which is a very good documentation of plant, animal, and mineral materials of India. So these are some of the works we are doing. And recently we have started a national mission called Swastik, Scientifically Validated Indian Traditional Knowledge. So today I'll mostly talk about the Swastik initiative. And this is the journal. If you want to know more, you can visit our website. So in Indian Journal of Traditional Knowledge, we cover multiple domains like traditional agriculture, animal husbandry, architecture, food, handicrafts, Indian system of medicine, other relevant domains also. So it's a multidisciplinary journal. So you can explore this. Now talking about the promotion of Indian traditional knowledge, we know the dissemination of Indian traditional knowledge has multiple challenges. One is the lack of documentation. So like our grandparents, they were very rich in traditional knowledge, but they were passing on the knowledge orally. There were very poor documentation. So we lost lots of information because of this oral transmission mode. Then misinformation. What happened in traditional knowledge is, these days, everybody is using social media. But even today, even now, I think many of you must be using in your mobile because lectures are a bit boring. So the problem with social media is anybody can share anything. So this has happened in Indian traditional knowledge also. Everybody is sharing some information. Most of the informations are not correct. Like open Facebook, you will find lots of pseudoscience information, lots of wrong information. So when people get repeatedly wrong information, they think that this domain, the traditional knowledge, there is no science in it. Because of this huge misinformation, so people are losing trust on traditional knowledge basically. Then the knowledge holders, because of globalization, they are migrating to different places and we are losing the TK. There are unawareness also. So we don't know how rich India is on traditional knowledge. So I started this mission 3 years back. I'll be honest, before starting this mission, even I didn't know that we have this much traditional knowledge in India. Then lack of the younger generation. So younger generation is not interested. I can take example of food. We all know that our home-based foods are rich in macro and micronutrients. It's a wholesome food. But we very easily get attracted by the junk food. So that's one example. That happens with our building designs, like we are losing our own traditional architecture, our clothing and everything. So the new generation, they are a bit reluctant on traditional knowledge. Lack of integration in modern education. So when we studied in schools and colleges, we had no traditional knowledge in our syllabus. Luckily, since national education policy, traditional knowledge is a part of our education. So I'm also working with NCERT for making the NCERT science textbooks. Even these days, the NCERT textbooks, if you look at the class 6, 7, 8 books, they have some traditional knowledge examples in that. So now students are studying, maybe at later point, we'll have greater confidence and knowledge of the students and researchers on traditional knowledge. The legal mechanism of protection of traditional knowledge is also not very strong. There are not much effort to validate. Like, we know we have this much traditional knowledge, but scientific studies were not that common. So that was another issue. And overall in science communication, India or many nations, we are a bit poor in science communication. In traditional knowledge communication, we are even poorer. Like, we are not doing anything to promote the traditional knowledge. So, like, today we are doing this conference. So, some few scientists are talking to some other scientists. So, this knowledge is revolving in the scientific community. But to have that proper impact, we need to go to the society. They should know the correct information. So, this dialogue is very important. But now, the duty of every one of us is to proliferate whatever we have discussed to the society. So, that is science communication, and we need to focus on that aspect. So my mission called Swastik, this is started as per the suggestion of our Honorable Prime Minister, Narendra Modi ji. So he is the president of CSIR also. And in one of our society meeting in 2021, he suggested that we should study the traditional knowledge and what are the scientific evidences available. And if we find the scientific evidences, we should disseminate it to common people. So on his suggestion, we started this mission. As I mentioned, we have multiple domains of science where we have rich traditional knowledge. So these are some domains where we are doing the dissemination now. So from mathematics, metallurgy, chemistry, foods, water, commerce, everything. Everywhere we have rich traditional knowledge. I'd also like to declare that I'm not a very expert of traditional knowledge. My duty is to convey that information to common people. So suppose you people are traditional knowledge experts working in multiple domains. I'll talk to you. You give me the information. I'll simplify the information. I'll again share with you. You will verify that this is correct. Now this can go to people. Then I'll disseminate. So we do that thing. And it's also not possible to be expert of every domain of science. TK is like that. The knowledge is in every domain. I'll know a bit of life science, but in mathematics, I'm not in a position to validate that information. So for that, I am taking help of mathematician. And with that confidence, I'm saying that this is a scientifically validated information. Because saying scientifically validated is also come with lots of risk. You have to be very confident. So we have created nationwide network for promoting the traditional knowledge. By the way, during this journey, you will also learn how you can start a science communication mission in your institute. So this will show you the multiple approaches which can be taken. So we established collaboration in these different subject domains. Until now, we have done disseminated 81 interesting stories on Indian traditional knowledge. All of these informations are on social media. We have disseminated the content in 19 Indian language. Why Indian language? So, as per one estimation published in Nature Group, only about 10% people in India can understand, read, write. They're comfortable in English. So, majority of our population, they cannot understand English. So, if we do everything in English in science, then what about those people? So to convey our information, we must do dissemination in our languages. So that's why language is one aspect of our mission. This is my suggestion to all of you also. So when you can become a scientist or a lecturer, but you can do parallelly science communication for your society. Like you don't have to be a full-time science communicator like me to do a science communication thing, but it can be done parallelly. But when you do your science communication, please do in your own language. Like here, you can do in Marathi also. You do in English, but then translate it to Marathi and then put it on social media that way. So it should go in your own language, then only it will break that barrier and then reach the common people. We are doing in 5 foreign languages also, German, French, Russian, Spanish, and Japanese. Why in foreign? Because we want that our Indian knowledge should global also. So because of that, we are translating and we are disseminating. I'll give some examples of what kind of stories or what kind of rich traditional knowledge we have. So I'll touch like some domains, but because of the time constraint, it's not possible to cover everything. So starting with metallurgy, this is metal mirror of Aranmula. Have you heard of the metal mirror? We all know glass mirror, right? So there is a village called Aranmula in Kerala. They make mirror out of metal. It's a copper thin alloy. And they do fine polishing. So this is not glass, this is metal. And this mirror gives better image than our normal glass mirror. So before doing this mission, I didn't know about this mirror. So this kind of technology we have across India. So both these are done, studied by Sharda Srinivasan. She's a Padma Shri, from NIAS, Bengaluru. She gave us these studies, and lots of scientific studies has been published on these two aspects. So this metal mirror we have disseminated. The second one is wood steel. So this is a high carbon steel. So in normal steel, we have about 0.5% carbon. In wood steel, we have 1 to 1.5% carbon. India was very rich in wood steel production before British came. So in 1857, whatever has happened because of that, they have destroyed all our weapons and then weapon making technology. So we almost lost this wood steel technology during that time. But wood steel again, so we used to export this, and then it's also famous as Damascus steel. So many of you might have heard about that strong Damascus sword which was used at that time. So this is the example of that. So then this Ramappa temple. Any of you know about the Ramappa temple? So these days we are like constructing lots of building, and then our expert architects, they do lots of studies, they do it, and then we know what's the age of our building, like 90 years, 100 years, not more than that. So this temple is 800 years old, and it has resisted multiple major earthquakes. So how this is possible? So like 800 years before, we had this kind of technology which can resist earthquake. So it uses technologies like sandbox foundation, and they use bricks called floating, like it floats, so floating brick technology, and other careful material selection and all. So with that, they have this, they built this temple, and this is a UNESCO World Heritage Site also. This is just one example of Indian architecture. We have hundreds of examples like this. So we are that rich in Indian architecture. Then coming to Sacred Grove, so this is a bit relevant to today's discussion. So that year it was so common that everywhere, and then now having millet is becoming a fashion. Even one of our lab, CSIR CFTRI, they have designed one multi-millet bun, and they are like promoting it with the help of McDonald's. Globally also, the millets, they are now being celebrated. International Year of Yoga, we know. There are lots of scientific work also. If you look at the scientific publication on traditional knowledge, a huge fraction of them is on yoga and meditation. Very well impact papers. Very recently, WHO had organized the second global summit in Delhi. So that was also a common, like very positive development for TK. Coming to our food habits. So when Indian food habit is very it's very rich, very scientific. So we have this, in Assamese we call it poita bhat, multiple like panta bhat. So whatever is the leftover rice, you keep it and then you add some water, keep it overnight and it's consumed the next day. I think in different places of India this is consumed. It has different names. But that was just a traditional food. But when they studied it scientifically, they have found that it has probiotic bacteria. So you don't have to buy probiotic separately. In your traditional food only, it's rich. We just need to study it and then we just need to inform the common people. Then when they studied our food, they found that the carbon footprint of Indian food, it's lesser than multiple other countries they compared. So we are like...
Sorry to interrupt, sir, but we are running out of time.
Okay. I'll just finish in 2 minutes, sir. So we are meeting that carbon neutral guidelines also through our food habits. And then if you are looking for research opportunities, so DST has a cell called SRI, Science and Heritage Research Initiative. And then Ministry of Education, they have one IKS division. So both these, they are announcing for project calls. You can like apply for research projects and they support those things. So if you want to take research projects, then you can visit SRI and Ministry of Education's IKS division. These are our Swastik platforms. We are on Twitter, Facebook, Insta, LinkedIn, YouTube. So follow us and then help us to amplify the traditional knowledge across India. We are open for collaborations also. So if you have a very good story on Indian traditional knowledge and you have sufficient scientific evidences, we can help you to take that story to a global stage in all languages possible. And then you can contribute stories, you can help us in translations, so multiple others. So if you are working in any of this, then you can write to me. So thank you.
Thank you so much, sir. The session is open for questions. Any questions? Thank you so much, sir. I request Dr. Vishwa to present felicitation to the speaker. I request Dr. Aparna Pat to felicitate the chairperson.
The next speaker is Dr. Kishore Mohanta, Assistant Professor, Department of Applied Biology, School of Biological Sciences and Technology, Meghalaya, USTM, Meghalaya, India. Leader of the DST ANRF funded Nanobiotech Track K Lab 2022-2024 and Z Genza Malta Research Networking Project. Dr. Mohanta pioneers biofunctional nanomaterials, nano antimicrobials, and nanonutraceuticals from natural resources for food, agriculture, medicine, and environmental sustainability. A prolific researcher with 124 plus sci-fi articles, 35 book chapters, 4 Indian patents, and 10 edited books. Ranked in Stanford's top 2% Global Scientist 2025. Chief Coordinator for Microbiologist Society of India, Northeast, recipient of Education of Sustainable Development Award 2024 and Young Scientist Award 2025. He bridges nanobiotechnology, microbiology, and sustainable development. Please welcome Dr. Yugal Kishore Mohanta.
Thank you.
I request Dr. Girish G. Poddar, Professor and Head of the Department of Botany, Yashwantrao Chavan College of Sciences, Karad, to chair the session.
Shall I start? Good afternoon, all of you. Namaste, Karad. So, thank you, the organizing team, your Krishna Institute of Science and Technology, Krishna Vishwavidyapit, Karad. This is my second visit. The first Biomake in India. First, I was here. Second, I was absent. And in third, again, I am here. So it is a privilege for me to be here. And especially I thank to respected Pathade sir, because he has called me first regarding this invitation. Anyway, I am also thankful to Dr. Burman. He has made many things very simpler for my presentation because many things will be matched. So I will skip all those things. So when I think about Indian traditional knowledge or Indian knowledge system, so though I am working on nanotechnology, nanonutraceuticals, but when nowadays the need for to revive, to know, to spread the Indian knowledge system. And I am staying in the northeast part of India. All of you know the beauty of northeast India, the diversity of northeast India. So I want to get into a virtual tour of northeast India indigenous knowledge system. So now we are living with ultra advanced technology era. And again, we are talking about traditional knowledge system. So if you think these are both are contradictory, but fortunately or unfortunately, we have to think about the need of the hour. And need of the hour through the Sustainable Development Goals by United Nations, we have to think Indian traditional knowledge, we have to imbibe the Indian traditional knowledge, not only through the knowledge, or imbibe the knowledge in our mind. Rather, we have to practice in our daily life. What Professor already like Agnihatra and all, Havan, we have to practice to keep ourselves peace, healthy, and make the peaceful society. So what the urgent need for to balance ethics, sustainability, and humanity. You see, India is now also struggling for achieving different sustainable development goals. The 17 sustainable development goals, you all of know about it. And we are all struggling because you see, now also in red zone, India is inside a red zone, major challenges still remain with us. Though we have now achieving more rapid technology development, but still we are remaining with the challenges. Means we have to think about our traditional system, our traditional knowledge, make them scientific and implement scientifically, and we have to practice in our day-to-day life. Here you see now the country rank or India rank in Sustainable Development Goal achieving is 99. So still we are struggling. You see no poverty, we are achieving or we are progressing, and reduced inequalities. We are just achieving. Otherwise, other Sustainable Development Goals you see, we are still struggling. And now India is in the 4.39. It's if the 0 to 10 best score, India's score is 4.39 by 2024, and 2025 result has not come. So if you see good health and well-being, the part of the conference theme, still we are struggling. You can see in India the red mark. So still we are struggling, and we have to revive from these struggles through our Indian knowledge system, our rich Indian knowledge system. So bioprospecting Indian knowledge system or indigenous knowledge system of Northeast India, as I am staying there, that's why I am trying to present Northeast India in front of you, how people are living there, how they are still practicing some traditional knowledges. Because you know the more than 200 to more than 500 different indigenous groups are still residing in Northeast India. So they have their own practice from food, agriculture, textile, everything. They have their own practice of each individual indigenous group. Though they are staying in a single district, but if they are 4 to 5 different indigenous groups, they have different practice of cooking, different practice of consuming rice, making different vegetables, consuming your leafy vegetables, even different non-veg items like this also. So traditional food system of Northeast India, the most diverse traditional food system, if you get, then you will find in Northeast India. So what are the values of traditional food system? All of you know, we need a sustainable sourcing, wild or underutilized species that thrive in the local climate, promoting sustainable development food system and income stream for local communities if commercialized responsibly. Traditional knowledge system possesses extensive generation-old knowledge about the medicinal and nutritional use of local flora and fauna. And ethnobotanical wisdom serves as a crucial guide for modern scientific research to identify promising candidates for new nutraceutical products. They have knowledge, but though they belongs to different indigenous group, they have different languages also there. And as per the Dr. Burman already stated, that all that knowledge resides within their own language, within their own indigenous group, and they just transfer them through oral method. They are very less documented. So now need, as per the Indian knowledge system, we have to make it more enriched. It is already enriched, but by practice now we are not enriched. So with the modern generation, what Gen Z you can say, or we are here, we have to think, we have to practice those Indian knowledge system, and we have to adopt in our life. Probiotic potential, you know, in all fermented products, fermented food, fermented drinks, you definitely will get probiotics. And their food system is enhanced nutritional value, definitely they have, because wild edible plants, fruits and fermented products are more nutritious than their conventionally cultivated counterparts. And rich source of bioactive compounds, you know, when we consume any leafy vegetables, plant materials, definitely they have different bioactive compounds like flavonoids, alkaloids, and have their activity like antioxidant, anti-inflammatory, antimicrobial, anti-diabetic. Many of our researchers now working on this particular field. And importance of indigenous people's food system, health and well-being, the one part, spirituality, because many things they are adopting, they are consuming, or they are practicing only virtue of their spiritual practice. Culture identity, the food system, their dressing or the costume or their cloth, their way of cooking, these are all reflecting their unique cultural identity. Food security is the part, we are also struggling for that. And climate resilience. According to climate resilience, they are thinking about their own environment, and accordingly they are taking care of the different plant diversity. And environmental stewardship. It is the most important nowadays. Indigenous people's staple food system, rice, in that part, even throughout the India, we are all taking rice. It is very common that we are consuming rice. But when I consider that these 7 states of the northeast part, you see they have also different type of recipes of rice. In Assam, more than 500 different varieties of rice are available. Sticky rice, aroma rice, like this black rice. In Manipur also, well known for the black rice. And Tripura also, aromatic rice. So they always cook in different way. In Assam, the people cook rice in banana leaf. The sticky rice they are cooking in the banana leaf. And in Meghalaya, you will find, especially in Khasi communities and all, they cook rice by using bamboo. They take the bamboo, and inside the bamboo they put rice and go for boiling. Like this, they consume rice. Manipur also sticky rice, steamed, and black rice they consume. So though rice is a staple food, but they consume in a different way. That is also a part of their indigenous knowledge. Protein food source, definitely we are the chief source of protein. We always consider the non-veg system. And there, if you consider the non-veg system, they almost consume everything. Beef, pork, fish, lamb, whatever, chicken, everything they consume. Whatever the availability is there, they consume all sorts of non-veg items. But they also cook them in different way. Sometimes they ferment fish also for a long time, and sometimes they use bamboo to cook any fish or chicken or pork items. And specifically in Arunachal Pradesh, is mithun smoked, mithun, the animal, they take the mithun for a very delicious non-veg source. So they used to dried, boiled, fried non-veg items. Assam, fish, chicken, pork, everywhere you will find most Assamese people, they definitely take this fish, chicken and pork as a non-veg source of proteins. Nagaland, pork is very popular. Fermented fish, different insects they consume, silkworms, grasshoppers, and frogs also they consume for medicinal purpose. So unique flavors, because if you see the same thing they are consumed, but they prepare those food items in different way by addition of different flavors and make them unique flavors for their different groups. So if you see the fermented soybean, the soybean they ferment, but in different way they ferment so that they have put different name also. Tungring Bai, it is known in Meghalaya. The same fermented soybean, Akhuni, is known in Nagaland. And Mizoram also, Sattu, Manipur, Nagri, Arunachal Pradesh, Pehak, they consume this, your fermented soybeans. But Tripura, very less people consume, but major they consume the local herbs and bamboo shoots. Bamboo shoots are very commonly found, and they add every vegetables items, they consume, this add bamboo shoots. Assam also local herbs, chilies and mustard, khar, a unique alkaline liquid from banana peel ash. They burn the banana peel and form the ash, and they consume with their dish. And in Nagaland, Akhuni, the fermented soybean, bamboo shoots also they consume as a fresh and fermented. King chili, known as Bhut Jolokia, is a common name of that king chili. Extremely hot, used for the flavor. And Thatu, the chutney, they take. These are very common vegetables, you can say, they consume for leafy vegetables, mushrooms, lotus stems and roots, they are mostly consumed in Manipur, they prefer most. Bamboo shoots everywhere and wild edible fruits. Not only this is the only one example of wild edible fruits, but there are many wild edible fruits they consume seasonally. And there are different traditional alcoholic fermented rice-based beverages and microbial assessment of Northeast is a study by the scientific study by one group, Loing et al., Journal of Food Biochemistry. They have published this paper. From that paper, I have retrieved this. So what the beverage they prepare and what microbes are association with that by culturing in the lab scale, they have studied, and what type of different plant materials they use, they have also reported here. So this is only few, but more informations are still within the indigenous people, which are not still now retrieved. So it is one photograph from traditional dish preparation by the missing tribe of Assam. This lady belongs to missing tribe of Assam, and how they do prepare their dishes. And it is preparing anunu, a typical Idu Misimi tribal dish. Idu Misimi, one tribes from Arunachal Pradesh. They prepare this one dish with fish and sticky rice during Reha festival. This is the first week of February, it's just coming. They are celebrating the Reha festival, where they definitely prepare this fish and sticky rice combination of food. So one success story, if I can say, from rhododendron wine already in a marketed condition, from rhododendron petals, and specifically that Mompa community, that traditional knowledge, that fermentation traditional knowledge from the Mompa community, and now it is nicely commercialized to the rhododendron wine, because rhododendron flower is very much abundant in Arunachal Pradesh, Tawang and West Kamen district. Another Lakadang turmeric. The Lakadang village is there in Meghalaya. There one turmeric is available, is known as Lakadang turmeric. And specifically this is grown organically by the Joyantia tribes. There are 3 tribes, Garo, Khasi and Joyantia, in major tribes of Meghalaya. And then Joyantia hills. And the tribes are differentiated as per the name, as per their hills. Garo hills, Garo tribe, Joyantia hills, Joyantia tribes, and Khasi hills, Khasi tribes. So it is in the indigenous, you can say the turmeric, known as Lakadang turmeric, and it's very high content of curcumin. Dr. Morvan, better you have studied all this thing. So more curcumin content, that's why Lakadang is now a prime light of research, and how the cultivation and their production can be enhanced. So it is a traditional knowledge, but now we will enter our modern science, our technology, our agriculture system, so that we can sustain this knowledge, their indigenous farming system, and enhance the production of Lakadang turmeric. And there you will find many underutilized edible plants, because means they consume many plants, they consume in everyday life, but still these are not available to many common people, means outsider or urbanized people. But I found this all underutilized, you can say, vegetables in the local hut or market. Every weekly, many tribal peoples, they bring these all vegetables and for the selling. And this is one wild edible fruit, Bacchauria ramiflora. And this is one form, sorry, this is wrongly I have typed here. Is a form, Diplasm esculentum, one form, that is edible and very tasty. When I shifted to Northeast for my job, and then only I came to know about the edible form, and I consumed, very tasty. But when you look, you will not look a very pleasant look. But when you prepare and eat, it is very tasty. This is saga item, where leafy vegetables, it is an edible form. Your Diplasm esculentum. It is one study published in Frontiers in Nutrition, that how Adi festivals, Adi is a tribe from the Arunachal Pradesh. Adi is a tribe from Arunachal Pradesh. And in different time, you see in different time they celebrate different types of festivals. So according to festivals, they consume different type of flora and fauna. They have calculated in their indexing, they have made indexing also, the how much flora and how much faunal part they consume in different festivals. So they, round the year, they celebrate different festivals. According to festivals, they prepare the dish and consume different vegetables, plant materials, and your non-veg items. So how they contribute means this food system, traditional indigenous food system or knowledge, contributes to sustainable development goals, supports local economies, small-scale enterprises, and marginalized communities, creating sustainable livelihood. And really, now also many tribal peoples weekly or twice a week, they bring all those wild materials, consumable wild materials to the nearest market. They sell, and even we are fortunate that luckily we get and we consume those also local vegetables, and they are sustaining their livelihood. Promote diverse diet through wild edibles, unique ethnic foods, and integrated farming, reduce reliance on monocultures. Traditional and fermented foods are rich in vitamins, minerals, and probiotics, improving health and combating malnutrition. We all know as a student of microbiology and biotechnology, but thing is that though we know the probiotics are there, any fermented food you go, you will get the probiotics. It's the thing, but thing is that there are different way of preparing different fermented foods in Northeast India that still need to be studied. So we have to enter as a being a scientific community with them and study scientifically those things, and we have to find out what are more nutritional value, more potential of any microorganisms are there, so that it can be laterally we can explore industrially. These are the three major tribes from Meghalaya, Khasi, Garo, and Jaintia. You see here the different attires. They are in different attires. Means there is individual attires, color of stalls or pagodi, you can say, or the dress they have. Definitely they have their own indigenous knowledge of textile, their weaving system, color from the natural colors they use, because nowadays machines come, so we just making many piracy, no. This, we are just see the design and we try to do in a mechanical, means in machinery way. But they have been practicing those things since ancient time. The threads, the silk, and the color from the natural colors also. So each color variant reflects their own indigenous knowledge on particular textile. Nagaland, you see 17 major tribes resides in Nagaland. And here also you see Konyak, Ao, Chakesang, Angami. These are major 4 tribes. You see their pattern of attires. Here also this attires reflects their own individual or indigenous knowledge of their textile. Color, you can say everything. Mizoram also, Luisei, Ralte, Paite, and Pai. You see, if they wear this type of attires, so you can easily identify, yes, who are there, which tribes they belongs to. Manipur also you see Maitai, Kuki, Manipuri Naga means some Naga people are invaded in Manipur, Ukhrul area. So some Naga cultures also there. So now you know all the Manipuri case, Manipur disturbance is for Maitai and Kuki only. Arunachal Pradesh also there are 13 major tribes, and they have Adi, Aptani, Nisi, and Mompa. They have different attires, and this reflects their own indigenous knowledge. Assam, Bodo, Missing, Karbi, Rabha, Tai Ahom, they have also different attires. Means they reflect and they give the message till today. What also the Gamusa about, Dr. Burman was discussing about that. So these are their own socio-economic cultures still give us a message and scope for study more. Because if they have derived everything from the natural color, Means they have the possibility with their area that some plants or something they are giving color, natural color. So we have to retrieve those knowledge. Traditional herbal medicine, it is a very known thing because we are all are always trying to shifting to the traditional knowledge of medicines. So Bhut Jalakya, Misimi Tita, this Misimi Tita, this one, Misimi Tita is specifically used by the Idu Misimi tribes. Black turmeric is there, this is a black turmeric, is another profoundly found in northeast India. Thalkuni is very common, Central Asiatica is very common, but we generally take raw Central Asiatica in the empty stomach, but they use like cooking, like sag we take, so they use like this. And in the Bhut Jolokya is one variety of chili only. It is very flavor and hot, and it is very tasty also. When you put it as a sabji and all, you will get taste. And this photograph you see Yanung Jamal Ligo, the Padma Shri awardee from Arunachal Pradesh for the traditional herbal medicine. She has been practicing for the long times, and now also just few days before I just contacted one of my scientist friend from Nist Jorhat. His mother is suffering from the cancer. After failure of all chemotherapy and everything, now he is just taking the help of Padma Shri Anung Jamao Lego for control of the cancer and all. So threats to indigenous people food system, smart Western education system, you know, and rapid industrialization, protected areas, urbanization and modernization. These four are now all we are struggling. We are not ready to leave this situation also, but still we want how we can again establish the Indian traditional knowledge in our practice. Research priorities, always we are in a conference podium, we are in a platform, and we are within a scientific community only we are discussing. But when we want to do something, definitely the 50% part or the majority responsibility with funding and all, we should involve those particular indigenous people only. Those who have quite educated or literate and all, we have to involve them during our any project, any type of projects we want to continue or we want to pursue, then definitely we have to involve them. Respect the value of indigenous knowledge and ancestral wisdom on the same level as science, and recognize indigenous people as equal expert. Sometimes we do a blunder mistake. Always we underestimate many people that you don't know anything, what you know, because we are the PhD holders, we are the professors and all. Many times we are underestimate. But now the time has gone. We have to consider them the equal expert. Means if we are organizing any conference or a workshop or something, definitely those type of indigenous people should be the equal expert, equal speaker, equal resource person should be here. Then only we can directly communicate what they want to need, what they want to tell, what they want to disseminate. Mainstream of the often overlooked indigenous perspectives and knowledge in conventional research and go beyond multidisciplinary to take a multicultural approach that blends indigenous knowledge and science, develop culturally appropriate research approaches with indigenous people that support indigenous methods of learning and knowledge transmission, like storytelling, observation, landscape work. I think this type of works, Dr. Burman, they are conducting in their institute. Respect indigenous people's rights as enshrined in the UN declaration, because we have to involve, means that's why I have given the title name inclusion. Pathway to sustainability, it's okay, we all are talking about the sustainability, but how much we are include them in our journey of sustainability. Understand community decision-making process and work with community institutions. Support power equalizing approach for indigenous communities and women, including inter-community networking and meaningful engagement in policy process. Contribute to the body of knowledge of indigenous people for their own needs rather than as an object of investigation. Research should facilitate learning and collective analysis within communities, and the communities should be the first to benefit from the knowledge produced. Then only they have the faith on us. Yes, if we disseminate their indigenous knowledge to us, if they will work with us, then definitely they should have the belief that whatever the output will come, that definitely their community first will benefit. So these are different interdisciplinary research methods, how we can grab the knowledge of indigenous people, involve them, include them, and we can disseminate our knowledge with them as a blended mode and get some constructive outputs. Successful ways of research action. So we have to decolonize the mind of indigenous people who have been always brainwashed to think their foods, their practice, their cultures are inferior. Many times we underestimate, linguistically also and behaviorally, sometimes we underestimate people. Debunk the notion that the research is only done by the school, means only research degree holders, qualified, only can do the research, not like that. We have to include them, though they don't have that degree, diploma, or any courses, they have any degree. So use decolonizing action research methods where research is led by the indigenous elders and authorities, guided by indigenous epistemic principles, so that what happens, their knowledge is our primary source. So once they will not disseminate any knowledge, then we can do anything. Use indigenous research methods that frame research using indigenous world views and values, recognizing the spiritual values of food and links to the land. And challenges are there, definitely. So we have to also overcome different challenges. Language challenges is there. This indigenous food system is threatened by policies that restrict the use of forest resources. Nowadays, many protected areas are there. They are not even easily invaded protected areas, and they can't collect that all their indigenous food supplies like edible plants or other plant resources. Sometimes dry land rice, wild millets and foxtail millet have mostly disappeared because of green revolution methods. And now again we are remembering those millets, and now as per the Dr. Burman, is a fashion, and now we are more attracted towards millet food systems. So likewise, we have to rethink, revisit their knowledge, document the one part, another we have to include them and disseminate those knowledge into the practical field. Ways forward, what we need to do, already Dr. Burman has discussed. So policy needs to protect the indigenous culture that maintain food diversity. Initiative to promote traditional foods in markets and ecotourism. Ecotourism also already started, and many places you go, they're providing different cottage, like you will sleep on the mud huts and all everything, and they are providing their local foods. But those things must be come to our urban area also. Then only we can say, yes, we are really want Indian traditional knowledge or Indian knowledge system. Rituals that sustain Indian food system, indigenous food system need support, because many food systems, they prepare many dishes, they prepare many plants, they conserve just for any ritual purpose. So those rituals should be supported, those rituals should be respected, so that they will be more attracted towards conserving and knowledge disseminating. Thank you, organizing team, Biomake in India 3rd, KIST, KBV, deemed to be University Karad, India, and definitely Microbiology Society India. I am the part of Microbiology Society India, so I am working for Microbiology Society India. So thanks to me also, and definitely thanks to organizing team, Krishna Institute of Science and Technology, Karad. This is my university campus. I should show you because some attraction towards the northeast, ever you want to visit, so you can visit my campus. This is campus of University of Science and Technology, Meghalaya. This is another, the same campus, another building. This is also, but everywhere in hill terrain you will find all buildings, right? And this is the gate of our University of Science and Technology, Meghalaya, USTM. Thank you for your attention and engagement. Thank you.
Thank you, sir. I request Dr. Girish G. Poddar to present felicitations to the speaker. Any questions?
Are there any questions from the audience?
Final plenary speaker, Dr. Gunjit Kaur, Associate Professor and Associate Dean, Career Point University, Kota, Rajasthan. Esteemed delegates, we welcome the final plenary speaker, Dr. Ranjit Kaur, Associate Professor and Associate Dean, Career Point University, Kota, Rajasthan, and Adjunct Faculty at Ma'am, you are audible? Am I audible?
Hello, Gunjit ma'am. Am I audible? Hello, ma'am. Am I audible? Gunjit ma'am, please unmute yourself. You are not audible. You are not audible, ma'am. Good evening, ma'am. Am I audible? Am I audible, ma'am? Am I audible? Am I audible, ma'am? Hello, Gunjit ma'am. Am I audible?
Yes. First of all, I want to
Excuse me, ma'am.
thanks to Dr. Suresh Bhosle sir, Chancellor of the University, Dr. Neelam Mishra ma'am, Vice Chancellor, Honorable Vice Chancellor of KME Institute, Dr. Pramoon sir, Principal Advisor, and the person who contacted me, Honorable Dr. G.R. Patwari sir, Dean, KVV Institute, and Dr. Deshmukh sir from Microbiology Society. The team gave me this opportunity to present in this International Conference of Biomake in India Conference 2026. The Krishna Institute of Science and Technology, Krishna Vishwavidyalaya. So first of all, this is my happy thanks to all of you, all the management committee to give me this opportunity to speak up in front of you. So first of all, I want to just brief about myself. I am Dr. Gunjit Kaur. I am an academic background of last 11 years. I did 2 years research work in liquid biofertilizer production in RKU project in Rajasthan itself in my university, in Maharana Pratap University of Agriculture and Technology. From there I had did my PhD, which was completed in 2015. So here I will just try to fit my topic in this theme, microbiology, biotechnology in traditional knowledge in the applied life science, agriculture and traditional technology, as I am faculty of agriculture, specialization in microbiology and biotechnology. So I have put in my title as microbial enterprise traditional wisdom meets modern biotechnology. How microbes, which are endocrite, which are here incorporated in the plants, are having their traditional wisdom, and by the help of new biotechnology techniques, we can use it for enhancement of the crops. Defining the endophytes from the ancient wisdom to the modern science. Yes, in 1866, scientific origin, German botanist Agronom coined the term endophytes meaning. Endophyte meanings within the plant to describe microbes residing asymptotically inside host plant tissue throughout their entire life cycle. As it is that in models, bacteria are there in the latent spots. So endophytes are those bacteria, beneficial bacteria, those are living inside the plants, within the plants. So this is the scientific origin, and it was originated in this is from 1966 in 1866. Presently, to make it with the global reliance, over 80% of the world population continues to depend on natural herbal products for primary health. Yes, we are using tulsi, ashwagandha, apart from this aloe vera and many other these days moringa powders are being used in many remediation products. And so we can say that representing an unbroken leaf from the previous era. So traditional use from the last years, approximately 10% of the vascular parts, so those are in the relevant foreign parts, have been utilized in traditional medicine system across cultures for the millennia, particularly in Asia, Africa countries, where indigenous knowledge system flourished. Key insight: endophytes primarily originate from the rhizosphere. Rhizosphere is the rhizobium area near the root system of the plants and all the latent plants. The nutrient-rich soil zones surrounding in the plant roots are colonized plants through natural openings such as stomata. So stomata will help to accumulate all the processes. So the minor nodes of the root emergence point. Second, the modern agriculture crisis. What are the modern agriculture crisis? Because by the heavy use of agrochemicals, by pesticides and chemical sites, dependency, environmental impact, carbon footprint crisis. Yes, we are not able to save the carbon point due to the heavy use of chemicals and pesticides. Agrochemicals production and application contribute approximately 4% of the global greenhouse gas emission, representing a significant environmental burden in the fight against the climate changes. Because these global greenhouse gases are responsible for the crisis of the carbon footprint, uptaking the carbon points in and indeterminate agrochemicals used during the soil acidification. By the using of these chemicals and pesticides, the soil of our ground is continuously acidified. Nitrate contamination of the groundwater and the microbe will dissolve it. That's why the plants are not able to get enough number of maximum number of bacteria, the beneficial bacteria. Those are converting the complex form into the normal form of the elements and the compounds. So, yes, I have tried, I have aligned all the point of my PPT from the LTG because nowadays all the universities have to align with the LTG goals. So this of my slide and point, this tells you directly to do LTG 12, responsible consumption and the production. So yes, we are only responsible how to consume the plant crops and to keep our soil healthy. So it places urgent reduction of hazardous chemical inputs and promotes the sustainable practice. The second one is the paradigm shift. Paradigm shift means how previous we can say the number of bacteria present in the soil area suddenly less down. The graph is shifted down from higher to low. So from individual to hologens. Hologens means it is a group of, or we can say a consortium of bacteria, all the beneficial bacteria. Plants are no longer viewed as isolated entities but as of microbial bacteria virus living on inside. Those microbes intimate and influence each other development, immunity and evolution. So they are in a symbiotic relationship with the plants. These are beneficial bacteria which are staying in the rhizobial sector. They are in the symbiosis with the plant roots. Modern biotechnology aims to create enhanced plant habitat through purposeful science-driven microbial invention strategy. Yes, when I have did this work 5 years back, so we had also I have isolated Azabacter strain from entire Rajasthan state, from all the state of the Rajasthan. And Azosporium also I have isolated. We have done all the biochemical tests, molecular tests, and after that some studies we have sent for the gene sequencing also. So that time we have made consortia also. So consortia for rhizobium type of bacteria individually, and for these asymbiotic bacteria like Azabacter, Azosporium, we made one consortia also. So the group of same type of bacteria are more impactful for the soil health to make them more healthy. How endophytes empower plants directly and indirectly mechanism. So direct growth, yes. So the points which I am already discussing and clearing that solubilizing essential micronutrients, phosphorus, potassium, zinc for enhanced bioavailability of the plants. So the plant can easily obtain these nutrients directly phosphorus. So from complex to simple form. Biological atmospheric nitrogen through specialized enzymatic pathway that is nitrogen available in the environment, and how the bacteria are fixing these nitrogen in the soil, and after that the plant roots are taking that nitrogen. Indirect biocontrol production of antimicrobial compounds and cytophores that sprinkle iron through straight competent pathogen. So this is the second point, how to save our soil, because in indirect method the bacteria are responsible or they are playing a role of immunity. They are not can multiply or replicate these bacteria when we are giving them a proper substrate. So easily we can multiply these bacteria. Those are friendly to the soil and they have a symbiotic relationship with the soil and the plants. So how can we use the modern biotechnology as a revolution omics science for the microbial factory? So as in the fermenter, how we grow these bacteria, these beneficial endophytic bacteria for the person. Omics refers to the large scale of biological field that study closely of the specific biological molecules. So omics is related to the biotechnology, so which we learn about genes, proteins, metabolites and the process in the organism to understand the complete data system. So here we have to unlock the omics toolbox for the endophile studies. Just like metagenomics, directly we are dealing with the bacteria without culturing them, as we are doing culture in the labs and we are growing the bacteria. So directly studying the genetic material genes of the bacteria which are available in the environment, water, transcriptors. So we will study about the RNA structure of the viruses and bacteria, those are beneficial. Advanced sequencing technology reveals previous invisible endophile profiles, uncovering their genetic potential and functional capability at unlimitation resolution. Yes, when we did that, I did that 1.5 year research. So what we found that I have explorated 192 azotobacter at that time. So out of those, 10 to 20 azotobacters found give their best results in the wheat and in sugarcane. I'm talking about the biofertilizers that we had made. After that, we found all the biochemical results were okay, positive in those 192 azotobacter strains. But the 10 to 20 azotobacter strains we found. So after that, we have sent them for the gene sequencing. So here we can quote that my life example that yes, after doing all the biochemical and molecular tests and the morphological tests, we can send those bacteria for the confirmation for the sequencing also. Functional trait identification enable precise identification to key traits such as atmospheric nitrogen fixation genes, PSB, phosphorus solubilizing bacteria for the pathway, and the antimicrobial biosynthesis clusters. So in clusters, we will use many bacteria. Those are the same characteristics and use. And endophytes are the microbial factory. Yes, in this picture you can show they have tried to show as a big fermenter, in which fermenter we are fermenting or we are making the replicate of the bacteria. So metabolite biosynthesis, endophytes synthesis mimic bioactive metabolite that can mix and even enhance the therapeutic compounds produced by their medicinal plant host. So by the help of these endophytes, we can increase the number of enzymes those are producing by the plants only. So sustainable, if we are talking about the modules in the rhizobium, by isolating those modules, the bacteria from the rhizobia, then after culturing them again when we are putting in the soil, mixing in the soil, we can increase the number of bacteria over there. So here we can say that how endophytes as a microbial factory. In this this one is related to the rhizobium area, but again when endophyte is a microbial factory, if we are applying that bacteria in a medicinal plant or we are making a symbiosis relationship with that, the plant increase the efficiency for the photosynthesis will increase, the production of secondary metabolites will increase, antioxidants will increase. So endophytes here we can prove that endophyte can be used as a microbial independent of the season and the geographical content. Yes, when we have applied this endophytes in the plants or the symbiotic relationship, then the plant will not depend on its season for the production system and on the geographical and the climatical change. The plant automatically will generate more alkaloids, more antibiotics when it will be treated by the endophytes. Facilitates rational design and engineering of the crop-specific microbial consortia tailored with the particular environmental condition and the agricultural challenges. Yes, by using this consortia to mix up the similar type of bacteria or hobulants word previously I have shared with you. So this will particularly can challenge the environmental condition and the agriculture challenge and tailor it, and the plant will definitely produce all the alkaloids and antioxidants. From accidental to purposeful symbiosis, the enhanced plant hologen definition it is a complex or we can say a consortium of bacteria. Plants are complex assemblage composed of closed plant genomes plus all the associated microbial symbiosis like bacteria. Hologen is not a single organism. It is not an individual organism, but rather a collection of different species of organisms, including in which host organisms are already there, bacteria, virus, fungi, and within and on the host organisms. Enhanced plant hologens represent purposefully established plant microbe association. So in rhizobium we can say that it is affiliated with the wood models. Designed to boost agricultural productivity, enhance stress tolerance and improve all over plant resilience. So in modern engineering, contemporary science transition from the experimental relationship to rationally designed system using selective and bioengineered endophyte strain for the precision farming. Bioengineering, maximum of participants and the listeners are from the biotechnology and microbiology. So we are using the bioengineered bacteria also. So we can use these bioengineered strains, endophyte strains for the precision agriculture. Traditional practices, indigenous agriculture methods like crop rotation, organic amendment, composting, uni-intentional forced beneficial symbiosis contributed a long-term soil health and sustainability. By using this traditional practice, previously we are using this char, buttermilk, and cow dung, and all the Jeevanmrit, Astar, and all these, and that time our soil is very healthy. So now, through this conference only, we are promoting that use all the traditional knowledge for making India eat healthy, our soil healthy. Bringing traditional and modern, the wisdom imported in traditional agriculture practice is now being validated and announced the molecular biology, creating a powerful synergy between the ancestral knowledge and the cutting edge of the biotechnology. So by the help of this biotechnology, genetic engineering and microbiology system, we can enhance our soil health and make the soil sustainable. Traditional wisdom medicinal plants hidden the microbial patterns. Yes, the vascular plant. What is the problem behind that? Yes, we know there is no break in using this herbal tea, herbal medicines, tulsi, ashwagandha, many of the aloe vera, neem, our old age person at home and we are also. I am using that moringa tea at daily basis. But what is happening in the vascular plants in medicines? Approximately 10% of the vascular plant species having approximately 10% of the vascular plant species having documented using the traditional medicine system dating back to millennium. So this we have to work. That only 10% vascular plants are searched out. So many of the plants are there on which we have to work. resources, producer and the information and for the stability. Hidden purpose living, traditional healers initiatives value on the specific side and soil where medicinal plants are cultivated. Modern science now reveals that the local specific endophytes significantly contribute to the medicinal potential through enhancement of bioactive secondary metabolite production. So this one is lag log in the log fields of the growth cycle secondary metabolites are producing. Modulation of plant biochemical pathway, independent synthesis of the therapeutic compounds in the medicinal plants only. What traditional healers mean empirically, the place matters. Modern molecular biology now explain mechanically through the lens of plant-associated microbial communities. So, by the help of biotechnology and by the help of microbiology, we can isolate the beneficial bacteria, the enterophytes from the soils, from the plants, and we can use it to replicate them in the plant and the soil for the beneficial production of the medicinal enzymes. Modern biotechnology, the omics revolution in the endophytes research. Endophytes research. Decoding the invisible microbes, originally advanced in the metagenomics, the environmental bacteria, transcriptors, the RNA strength, proteomics, the protein structure, function, and the metabolites. All the metabolites studies their functional characterizations enables a comprehensive profile of endophytes communities that were previously undetectable using traditional culture-based methods. So here I can quote my example of my research that what we have did, we have did the morphological study, biochemical study, and the molecular study of those bacteria. So yes, by the help of biotechnology, metagenomics, isolation of the bacteria, the study of their DNA and RNA, what are their protein study, in their cell how much protein is present, what are the function of those protein, and the metabolite study, what metabolites they are producing, in which metabolites they are using as an enzyme to increase the number of enzymes number, enables comprehensive profiling of it. So by the help of biotechnology and microbiology, we can isolate and we can multiply those bacteria. Understanding mechanism of action, direct growth promotion, and secondly indirect promotion. So solubilizing phosphorus, potassium, zinc, ZSD, solubilizing bacteria, PHSD, phosphorus, solubilizing bacteria, fixing the atmospheric nitrogen, the residual bacteria responsible for that, producing phytohormones like all these are these are carbon plant growth hormones, oxygen, cytokine, gibberellin. Okay, so indirect protection like synthesizing antibiotics, siderophores, hydrogen cyanide, and lyticarbonate actively suppress the phytopattern of the pest population. So indirectly, what these endophytes are doing, they are not allowing the bacteria to grow on the plant, or previously they are damaging or killing those harmful pathogens. Precision bioinoculates, development of target strains specific and microbial treatment designed to replace the harmful synthetic agrochemicals while making exceed agricultural productivity and crop quality standards. So what is the technology breakthrough? High throughput sequencing cost have decreased over by 99.9 since 2001. So the price is decreased. So now we are able to make first sequencing. Decategorize the access of omics technology and accelerating endophyte research globally. So on global level, it is essential to do research on the endophytes and making use in all the plants. Endophytes and microbial factories busy anatobotany and the pharmacology. So yes, and secondary metabolites are phasing in after lag phase, log phase, stationary phase. After the stationary phase, the secondary metabolites are produced. So endophytes modulate host plant biochemical pathway, significantly increase the production of high-value compounds including essential oil, phenolics, antioxidant, alkaloids, and terpenoids. So all these happening in the stationary phase of the plants. Bioactive compound mimicry. Certain endophytes can independently biosynthesis unique metabolites that mimics or enhance the therapeutic properties of their host plants, offering pharmaceutical potential without the plant cultivation. So yes, that's why in the plant tissue culture lab is also pharmacology, because through the native plant tissue culture we can directly go through on the stationary phase of the plant and directly make these alkaloids. The plant will skip that, or we can say easily skip a small cycle of lag and log phase, and finally the plant will make these alkaloids in the stationary phase. So, anatomic studies how people use plant forcing on the culture use, while pharmacologists significantly investigate these plants for the medicinal purpose. So, endobotany studies how people use plants. So, in endobotany, how people are using the plants for medicinal purpose or for eating purpose. Sustainable drug development. Endophytes serve as a renewable living factories for the pharmaceutical compound production, bringing traditional anatomical knowledge and with modern industrial biotechnology and the green chemistry principles. So this one is the current data. So 5 trillion, 5,000, yes, 5 trillion USD by the 2050. So this is 2050. It is required by the consumer of all the natural product according to the study. And yield enhancement potential. Endophyte inoculates medicinal plants shows up to 300% increase in bioactive compound production compared to the uninoculated control. So in the plants in which endophytes we have applied, so there is 3% yield enhanced only by the presence of these endophytes. Future vision. The conversion of the traditional botanical knowledge to modern molecular biology and sustainable bacteriology equation, microbial endophytes as a cornerstone organism for the 21st century agriculture, medicine and the environmental restoration. Endophytes powered agriculture, a natural base revolution for the sustainable growth. Harnessing microbial partnership to transform food security, climate resilience and the economic prosperity in the 21st century. Yes, this we can easily quote to sustainable for the sustainable growth. So in previous slide I have already told it 5 trillion USD demand essential oil market by the 2050. The global essential oil industry represents unprecedented growth opportunities never having happened in exist before. So this demand never had been asked previously, and yield increased by 14 to 37%. This is the current data in market. So if we are quoting this with the SDG alignment, LTC aid is decent work and the economical growth. So yes, by using this endophytes, we are working in a sustainable manner. And finally, what we are getting, the outcome is it is an economical growth without investing with the low investment. We are getting more herbal plants with more antioxidants and this essential oil for the herbal purpose and for the medicinal purpose. So this photosynthesis process is higher in those plants. The chlorophyll, the stomata are working, they are making more chlorophyll. So ionophytes ultimately boosting the photosynthesis by increasing internal leaf CO2 via their respiration process, improving nutrient like NP uptake, producing plant hormones, auxin, cytokinase for better chlorophyll and enhancing water use efficiency. I am talking about the plant in which endophytes are there. Root associated microbes increase soil organic matter, creating a long-term carbon storage reservoir. Reducing greenhouse emissions, as in previous slides I have told, 2% carbon reduction is responsible due to the greenhouse gases. Specific endophytes like trichoderma, ascarium reduce nitrogen oxide emission nearly by 50%. Natural based alternatives to synthesize nitrogen fertilizer cut the agriculture carbon footprint. So by the help of these endophytes, we are cutting the prices and we increase the number of carbon and reduce the number of carbons. Directly this point is also quoted with the LZ30 climate action. Endocrines are directly responsible and direct beneficial from off-scale natural based solution to deaccelerate the global warming by maintaining the agriculture productivity, environmental resilience and the food security. Abiotic stress resilience like endophytes produce ACC dominance. This is a biochemical test. We have ordered amino cyclopropene carboxylate test enzyme. This one is produced by the application of endophytes, and it is responsible for lowering the making downward stress induced ethylene during the drought period, or we can say in the drought environment. They enable plant survival against the high salt in the presence of high salt, extreme temperature, and in the less amount of water and water scarcity. Bioremediation purpose, many of the plants are there which are responsible for the breaking down of the harmful bacteria. So endophytes assist the hyperaccumulated plants in extracting heavy metals and persisting organic solvents from the contaminated soil. So these are helping to kill, to remove all the contamination by the heavy metals and the heavy solvent. So endophytes are also doing work in the bioremediation solution also. So feeding 9.7 billions by the 2050, endophytes provide a natural positive pathway to meet global food demand without environmental degradation and expanding agricultural footprint. So by, we can say, इसकी help से we can directly quote that it is a footprint of agriculture. By using this endophytes, we can, many farmers or all the agriculture students, can we increase the maintain the sustainable agriculture in our world or India. So SDG life from land, restoring degrading ecosystem, SDG 2 zero hunger. Yes, by increasing the yield production, we can also maintain our SDG 2 also, and by improving the yield of the products and the crops, so we can decrease the hunger also. Addressing global challenge, climate and sustainability, carbon sequestration, endophytes plants help us. We create increased root biomasses in the rhizosphere area, soil organic matter by converting the complex matter into the simpler matter, actively removing CO2 from the atmosphere and storing underground for the decades. Because the plant itself doing work in increasing CO2 itself in the plants and increasing the photosynthesis process, chlorophyll content and the stomata are well working in the plants which are in the endophyte. bioremediation, all the heavy metals are also degrading and removing by the help of endophytes, using endophytes to clean heavy metals, lead, cadmium, arsenic, and recycling the tolerance from the contaminated agriculture and the industry site. So, where the industry in the industrial area, where the heavy metals are there, we can plant these type of plant will be applied with the endophytes. So, the soil will be sustained and we can make the soil healthier by the process of this bioremediation. These endophytes will work over there to make the soil healthy. Future perspective and challenges. Integrated bioeconomy. So these are biogen, and by this biogen we can increase the economy of India. Convergence of agriculture, medicine, and industry to amplify application to support the circular, sustainable bioeconomy across the multiple sectors. So these are biogens, beneficial bacteria, and by these we can increase our world or India economy at a higher level and with a very minimum cost. So these are living organisms, and after multiplying them and after applying them on the for the purpose of medicines and making herbal medicines for agriculture purpose, for increasing the yield purpose, and for sustaining the plant in all abiotic and biotic systems and all worst conditions. We can sustain the plant, increase the photosynthesis activity, decreasing the CO2 in the environment. So this is the integrated bioproduct which is directly increasing economy. So precision microbial engineering utilizing synthetic biology and AI to design optimal endophytes for specific target functions. With the help of artificial intelligence, we can directly relate that which bacteria is will be separated with which separate. So customized microbial solution for the diverse crop. So particular, as we all know that azotobacter is working for mainly for the wheat and sugarcane crops, and for lighting crop rhizobium cultures are there. So by the help of this AI and AI, we can first search out that which particular bacteria is directly associated with the which crop. Regulatory safety framework, developing clear regulatory guidelines and robust safety assessment protocols for size advantage from the seedling to maturity. So उनकी जो size थी एक comparatively it is more big, maintaining benefited even when compared to the standard chemical pesticides. So when the maize was treated with this endophyte to consortium of these consortium of these two endophytes, what they found that the pod size is higher and the yield began earlier. Essential oil in Thymus vulgaris, boosting secondary metabolic production in aromatic plants for the essential oil ester. What are the application mode? The whole inoculation of Bacillus lymphocaceus and Bacillus thuringiensis strains enhance growth in the essential oil production in this Thymus vulgaris, even in the complete absence of the traditional chemical fertilizers. So yes, currently what we are doing in my School of Agriculture of California University, my entomology faculty has made biopesticide as per the master and what it is a practical example. So I just want to share and quoting this by this point that absence of traditional chemical fertilizer. So we have turmeric, we have made turmeric and on broccoli. What we have did, we have not applied any pesticide and any fertilizer on that, and we have only applied the biopesticide. And what we found, the plants on which we have applied this aster, the plants are totally disease free, no pests were infected with those plants. So this is just 2 months back I am sharing this example. So my students of first year in this skill enhancement courses of first year, they by their own on hands-on training they prepare those aster, master, and we have applied in our top cafeteria, and we found those live results that the plants are no more the plants are disease free and pest free. Pharmaceutical and disease control application is study 4. Anti-inflammatory potential. Plant Emilia Sonfacea extracted from endophytes bacillus substrate results high anti-inflammatory activity
Hello.
in vivo.
Hello.
Significantly reducing inflammation
Excuse me, ma'am.
from model possessing endophytes and independent pharmaceutical source.
Excuse me, ma'am.
Second case study in wheat and barley production. Plants critical esteem, wheat and the holiday vulgus barley, bacillus subsistite.
Hello. Excuse me, ma'am.
Yes, ma'am.
Ma'am, we are running out of time. Please wind up the session. We are running out of time. Please wind up the session.
Yes. Okay. So one more example. So this one, this is concluded against the powdery mildew disease by the Anapodis endophytes, and the yield will also increase, significantly improving the iron phosphorus uptake also in wheat and barley by the using of biophytes. So one more example of sugarcane growth promotion. Target plant was sugarcane, saccharum octans, critical biofuel in sugar growth. Microbial strain was Panatena agrolia 33.1 and Acetobacter. Mechanism direct atmospheric nitrogen fixation and production of growth promoting phytoauxins. So by the help of these, what they will enhance plant biomass, total nitrogen content, and dramatically reducing the dependency on the industrial nitrogen fertilizer. So by using this bacteria, the microbial state, they have no use of extra nitrogen. So this one is directly related to climatic action SDG 13. Secondly, metabolite with ashwagandha, which was also. So using these two enzymes like minerosides and vinalpha for the active medicinal compound responsible for ashwagandha therapeutic efficiency, which is helping in many diseases. So it is by using ashwagandha or its enzymes, SDG we can put for good health and well-being. Environmental bioremediation, this one we have already discussed, but this one is an example of case study on tobacco diminution breakthrough result, what the result was that plant thrive in toxic soil while actively extracting pollutants and transforming unusable land into the productive ecosystem. So by this we can sustain with the life on land SDG, chickpea and legume enhancement, microbial discovery, bacillus substyle, mechanism of action. These enzymes produce ammonia. So the bacillus substyle is producing ammonia for nitrogen availability and cidrophore for the iron chelation. So enhancing mineral uptake efficiency. Pathogen suppression indirect antagonism against the root rotting pathogen through the competitive exclusive and production of antimicrobial metabolites. So finally what happening in this case study in chickpea and legume, significant improvement of both biomass of the bacteria and the seed production, ensuring the higher yield in the crop pods, protein rich crops for human consumption. Integration, innovation and action. Modern biotechnology does not replace traditional agricultural wisdom, it validates, amplifies and scales the centuries of empirical knowledge through precision tools and the scientific rigor. So by this we can maintain the food security by 15% project. We have to omix integration by using this biotechnology and natural approach, and by this carbon farming policy. So making credit for making these are carbon neutral. So what is my submission to this presentation that what is the call of action? All policymakers, researchers, agricultural stakeholders must collaborate to shift this chemical dependent system to the microbial based system MBS. The condition ensure long-term environmental sustainability, human health protection and economic resilience in the face of climatic change. Endocrine biotechnology represent the cornerstone of One Health. So by this only One Health we can at 360-degree angle health, human, we can cover all the living organisms, integrating plant, soil, human and plant health into the unfined framework for the 21st century. Yes, thank you.
Thank you, ma'am. Now the session is open for questions. Any questions?
Any questions there?
Sir, are any remarks from your side?
So at the end of the session of this Dr. Gunjit Kaur from Career Point University, Rajasthan, I am thankful to her for a very informative and detailed lecture about the details about the plant endophytes. So on behalf of the Krishna Institute of Technology and Science, I am thankful to Gunjit Kaur. Similarly, I am also thankful to all the audience, the students and all the participants for the active participation and the discussion. Thank you. And I am also thankful to the organizing committee for giving me an opportunity to chair the session. Thank you.
Thank you, sir. I further invite Dr. Jayesh Naure to acknowledge the session. Thank you, sir. Thank you, ma'am. Applause for today's luminous. Gentlemen, on behalf of this organizing committee, heartfelt thanks to the all speakers, session chairs, delegates and participants for making day 2 truly memorable. Your contributions enrich our theme's pursuit and sustainable harmony between ancient wisdom and modern progress. Thank you. Now we are proceeding for the alumni function after 10 minutes. It's a request to all the alumni to please to be seated in front. It's a request to all the alumni to acquire the front seats. All staff, students and the alumni are requested to be acquire front seats.
पटकन ओपन कर
start.
हॅलो हां सर करू देत ना मॅम बंद all the teaching staff, non-teaching staff and the alumni please acquire the front seats. Student please. सर्टिफिकेट द्यायचे होते त्या बोलू हां कुठे तिकीट बोलवा देतो तयार आहे घेतोय या म्हणा लगेच ना before we are moving forward alumni meet we are going to felicitate our esteemed juries for oral presentation poster presentation and model presentation. First I would like to call upon the stage Dr. Komal K. Bise, jury for poster presentation. I would like to call upon the stage Dr. Aparna Pathade, Associate Professor KIST KBBDU Karad, to kindly felicitate the jury. Next, Savita D. Mari. Our next jury is Vrishali Vishwarao Patil, Vivekananda College, Kolhapur. Thank you so much, ma'am, and sorry for the inconvenience.
स्टेजवर कोणी कोणी बस पठाडे सर, देशमुख सर. देशमुख सर. Good evening, all of you. I kindly request Dr. Pathade sir, Masurkar madam, A.M. Deshmukh sir, Jayan Pawar sir to be seated on the stage. Datkhele sir. शेवटचं कोण आहे? जयंत पवार सर. तिथं आहे का कोण? तिथं आहे का कोण समोर? आहे का? Good evening, everyone. A very warm and heartfelt welcome to all our respected guests, alumni, faculty members, and dear students. Today is not just a gathering, but it is a celebration of memories, milestones, and a bond between our institution and its proud alumni. To begin with this beautiful reunion, on an auspicious note, I request our respected dignitaries to kindly come forward for the lamp lightning of the ceremonial lamp symbolizing knowledge, hope, and enlightenment. I also request Dr. Aparna Pathade ma'am to enhance the grace of this ceremony by performing Saraswati Vandan.
जय शारदे वागीश्वरी, जय शारदे वागीश्वरी, विधि कन्यके विद्याधरी वागीश्वरी, वागीश्वरी, जय शारदे वागीश्वरी, ज्योत्स्ने परी कांती तुझी, ज्योत्स्ने परी कांती तुझी, मुखरम्य शारद चंद्रमा उजळे तुझ्या हास्यातुनी, उजळे तुझ्या हास्यातुनी, चारी युगांची पौर्णिमा, तुझ्या कृपा कृपेचे चांदणे तुझिया कृपेचे चांदणे नितवर्षू दे अमुच्या शिरी नितवर्षू दे अमुच्या शिरी वागेश्वरी जय शारदे वागेश्वरी वीणेवरी फिरता तुझी वीणेवरी फिरता तुझी चतुरा कलामयंगुली चतुरा कलामयंगुली संगीत जन्माये नवे संगीत जन्माये नवे जडता मतीची भंगली उन्मेष कल्पतरुवरी उन्मेष कल्पतरुवरी बहरून आल्या मंजिरी बहरून आल्या मंजिरी वागेश्वरी जय शारदे वागेश्वरी.
Thank you, dignitaries, and thank you, ma'am. Now, we move towards the floral welcome and felicitation of our esteemed guest and express our respect and gratitude. I kindly request Dr. G.R. Pathare, Dean, Krishna Institute of Science and Technology, Krishna Vishwavidyapith, to felicitate our today's chief guest, Dr. A.M. Deshmukh, President, Microbiology Society, India. Now I request Dr. Snehal Masurkar, ma'am, Dean Academics, to felicitate our Dr. Pathare. Now I request Dr. Jayshree Nanavre to felicitate Dr. Masurkar, ma'am. I request Dr. Abhay Gatke sir to felicitate Dr. Jayan Pawar sir, Assistant Professor, Krishna Institute of Science and Technology, Associate Professor. Abhay Gatke to felicitate Dr. Kailas Datkare sir, Associate Professor, Krishna Institute of Science and Technology. Thank you. Now I invite our guest of honor, Dr. Snehal Masurkar ma'am, to deliver the welcome address.
Good evening, delegates on the dais of the dais, my dear colleagues, and the alma mater of Krishna Institute of Science and Technology. A long way going behind 2007, we commenced with the college. We started with Krishna Institute of Biotechnology and Bioinformatics. Since inception till today, there were many ups and downs with the programs, with the schedules, with the different types of struggles that we have faced in the college, and then the students passed year by year. Now the position is, we remember the students only by faces. Sometimes we are not able to recognize the student because physical changes, or maybe the student reaches us after so many years and it becomes impossible to just, you were our student like that. But looking at Pathare sir or Deshmukh sir, it feels like there should be a good attachment of student-teacher bond till our last or so many years after we are meeting each other. When we look at Pathare sir also, many times it happens that sir says, he's my student. I think he was my YC student of so and so year batch. So I think the student and teacher relationship should be in this manner, that we should be able to recognize each other years together. Even the student should keep the respect of the teacher till whatever the relationship may continue. So it feels very proud to be a part of Krishna Institute as Science and Technology म्हणण्यापेक्षा कृष्णा विश्वविद्यापीठाचा भाग असण्याचा खूप आनंद होतो. खूप रिस्पेक्ट वाटतो. मुलं जेव्हा आपल्याला परत येऊन भेटतात आणि ते म्हणतात की मॅडम ह्या बॅचला आम्ही होतो किंवा तुम्ही आम्हाला हे शिकवलं. खरोखर टीचर असण्याचा तेव्हाच आनंद वाटतो जेव्हा एखादा विद्यार्थी आपल्याला नंतर येऊन सांगतो की हा चॅप्टर, हे टीचिंग, हे तुमच्याकडनं मिळालेलं जे आहे ते खूप मौल्यवान आहे. सो दिस इज नॉट अ फॉर्मल मीट और दिस इज नॉट अ मीट विच वी आर प्लॅनिंग टू, बट इट happens to be when the teacher looks forward to see his or her students to fly in the sky when he passes out from the institute. So thank you for the organizers to give me an opportunity to speak in front of you, and all the alumni who are present over here for their organization of the event and giving the respect. Even I am an alumni of Krishna Institute because I have passed my PhD from Krishna. So I am also a part of you, and thank you so much.
Thank you, ma'am. Now I request Dr. Jayant Pawar sir to address the gathering.
Good evening. Respected dignitaries on the dais, Honorable Dr. Arvind Deshmukh sir, the person behind all the meaningful collaborations. I always looked after him, how he's energetic at the stage and go to various places across India and bring good people. And not just India, but he goes abroad and take a good collaborations to KVV. The vibrant dean of the KVV, Dr. G.R. Patade sir. The energetic dean academics, Dr. Snehal Masurkar ma'am. Epitome in research, Dr. Kailash Datkere sir. All my colleagues, students, and of course the alumni gather here today. It's indeed a privilege to stand before you and interact with you. I always remember when I attend such programs, when I go to my institute as an alumni or in any capacity, I still feel the nostalgic things, and I remember those days which we spent in classrooms, in the laboratories, and of course in cafeteria. Most of the times we remember in cafeteria, and the moment we realized, okay, this is not the place where we should spend our most of time. And after some times we realized, yes, the place where we should go and spend our valuable time is laboratory. Because there also we can have fun, right? Yes. So, like I always think, what's the role of alumni in anybody's life? One day you become an alumni, some of them are already alumni, right? What is your role to play for the progress of the institution? That's very, very crucial. It's not just a gathering, it's not just a get-together kind of thing, but to share your experiences post-academic excellence that you have achieved through that institution. So when you get this exposure and again come back to the institute from where you belong to, you are supposed to share all your experiences, all the challenges that you have faced, and how you overcome all that to all the students and obviously your colleagues, so that they can make necessary changes in them, come forward and contribute to the society in meaningful ways, right? So I think it's a right platform. It's your responsibility to actually connect to the institutions. You help us to bring meaningful collaborations for generations to come. Also, you can like tell our students that like what kind of challenges you're going to face in that particular domain, so that they'll be prepared themselves, they'll develop themselves, and definitely work for the betterment of society and for the institution. So I wish you all the best for your future. Thank you very much, organizers, for inviting me here to be part of this wonderful gathering. Thank you so much.
Thank you, sir. Now I request Dr. Kailas Dakre, sir, to address the students.
Good afternoon, one and all. Respected dignitaries on the dais and off the dais. I feel it's a proud moment for us to be here on the stage on the occasion of this alumni meet. I have some complaint with these PhD scholars and some of the junior students, because this conference is for students and all research scholars, but majority of them were not attending this conference because it's a majority of scientific session. They have good scope for attending and getting the knowledge from this conference, but majority of students are outside the hall. Because majority of them not attended this scientific session. Many of them, like the whatever sessions are there, so they are very much interesting and very much there was for learning. So these kind of platforms are for students or research scholars. So in this platform they can learn many things. Whatever faculties or researchers they have created research or they are doing research. But like we can learn many things from such platforms, like not from only a single conference, but if they go outside the many conferences. So these platforms are there for understanding the research, like what research we can carry out and what are the scope for but it's okay, but like I have some I observe some this problem like but it's nice opportunity or good opportunity for students to learn things like but from next time you might be attending these such type of like conferences or research talks. So from my side, all the best for all the alumni who are majority of them are from the same college. So all the best for your next career. Thank you.
Thank you, sir. Now I request our today's chief guest, Dr. A.M. Deshmukh, to please deliver the speech.
Alumni meet is very, very important event. Why? I will tell you. In 1988, I was in Japan. And in Japan, you know that in Japan people don't understand English. Not Hindi. Most of the people were speaking only in Japanese. And we were in search of vegetarian hotel. And we were not getting. So we were searching on the road whether there is a vegetarian hotel, but whom to ask? Because no one understands English, how to ask? Suddenly we found one group, and they seems to be Indian by their posture and everything. So we went near them and requested, "Sir, are you Indian?" One of them came forward, "Yes, we are Indians." We were so happy. तो मैं बोला कि सर हिंदी में बात करेंगे ना हम? वो भी खुश हो गए। हाँ, हम हिंदी में बात करेंगे। आप कहाँ से आए? तो हमने बोला कि सर हम महाराष्ट्र से हैं. वो बोले वी आर फ्रॉम महाराष्ट्र. सो आता मराठीत बोलू. Correct? I'm telling you affection whenever we are studying in the one college. Then we started in Marathi and I asked that group, where from you are? Because I was serving in YC College, Karad. He told, sir, we are from Karad. All group told that we are from Karad. This is a real event, I will tell you. Then I asked him, from which part? They told, we are from Engineering College, Karad. Then we spoke in Marathi that we are from YC College, Karad. And then within a fraction of a second, we become a friend. Why? Which attachment? In Japan, two Karadian from the same college approximately meeting together. What is the importance of alumni? Alumni have the same. Whenever you are going for example, in hospital somewhere in Mumbai, and the doctor is telling that I am from Krishna Vishwavidyalaya, you will become so happy, sir, I am also from KVV. That is the importance of alumni. And alumni has a very, very important role in foreign countries. Even in Ferguson College. I will give one example. From Panvel, you might be knowing Thakur is there, who is a MLA. He is having crores of rupees with him. He is a past student of Rayat University, Rayat Vidyapith. And every year he is giving 1 crore rupees donation to the Rayat because I am a past student of Rayat. That affection everyone is having. So today, I really appreciate all teachers for organizing this alumni meet. That you will not feel nostalgic whenever you are here, as Jayant Pawar sir has told. You will feel nostalgic whenever you will be in Singapore and someone will meet you from the same college. So, that is the importance of alumni meet. And today the efforts are only to realize you, to do something for our institute because institute has given you a degree. On the basis of degree, now you will have a path throughout your life. You are using that degree. And who has given degree? This college has given a degree, this institute, this university. So that is the importance of an alumni meet. So let us decide together today that whatsoever possible I will connect with my classmates. I will connect with my university students, maybe 2 years difference, maybe 3 years difference. But if you are from the same university, you will have affection. And that's why UGC also has given an importance to the alumni meet. And there is no other person in the world which will support this institute than the alumni of this institute. So, today, as a president of Microbiology Society, I thanks for giving me a chair. And all the faculties which are sitting on the dais, I know, they are doing too much efforts for upliftment of the institute, for upliftment of the different students in the various subjects. Even the teachers are also sitting. And I have seen from yesterday morning, continuously everyone is working and with a pleasant faces, because to work is a simple, but with pleasant faces is a very difficult. And they are working hard, and I wish all the best for all alumni students of this university. Thank you very much.
Thank you, sir, for your inspiring words. Now, we move to one of the most emotional and meaningful segments of today's alumni meet, and that is a felicitation of our respected teachers by their proud alumni. Teachers are true architects of our lives. The knowledge they impart, the values they instill, and the inspiration they provide remain with the students forever. Today is a special moment where gratitude meets respect and memories turn into appreciation. I now invite our alumni representatives to kindly come forward and felicitate our respected faculty members with a token of love and appreciation. First, I request Dr. Jayashree ma'am to felicitate Dr. G.R. Pathare sir. I request Dr. Abhay Ghadge sir to felicitate Dr. Snehal Masurkar ma'am. I request Mrs. Meenal Patil to felicitate Dr. Aparna Pathare ma'am. Meenal Patil. I request Mr. Mohsin to felicitate Dr. Shilpa Rulkar ma'am. I request Mrs. Swapna Jingge to felicitate Dr. Jayashree Nanavre ma'am. I request Mrs. Neelam Thorat to felicitate Dr. Abhay Ghadge sir. I request Mrs. Kiran Patil to felicitate Mrs. Ashwini Jadhav. I request Prajakta Ghadge to felicitate Dr. Rahul Sonawale sir. I request Ms. Pranali Pawar to felicitate Dr. Narendra Kumar Surenshi sir. I request Tejasri Bhagat to felicitate Dr. Wasim Bhagwan sir. I request Ms. Shivani Kadam to felicitate Ms. Priyadarshani Patil ma'am. I request Reshma Bhag to felicitate Dr. Dhanashri V. Patil. I request Shivani Guzar to felicitate Dr. Jayan Pawar sir. I request Tejaswini Kalandre to felicitate Dr. Kailash Ratkere. I request Shabikta Mumin to felicitate Dr. Chidambaram sir. I request Sonika Patil to felicitate Dr. Selva Muthukumaran sir. I request Naman Mishra to felicitate Mr. Yashwant Patil sir. I request Saili Jadhav to felicitate Mrs. Sneha Kulkarni ma'am. I request Vaishnavi Jadhav to felicitate Dr. Amar Mohite sir. I request Manisha Jadhav to felicitate Mrs. Prajakta Patre ma'am. I request Aboli Shinde to felicitate Mrs. Deval Patil. I request Payal Bangar to felicitate Rohini Chavan ma'am. I request Avdut Gare to felicitate Rahul Kapurkar sir. I request Sonali Jagtap to felicitate Sudarshan Gore sir. I request Tejasri Varute to felicitate Adik Rao Mote. I request Harshada to felicitate Yogesh Bakle. I request Nikita to felicitate Purnima. I request Gunjan Jadhav to felicitate Mrs. Deepali Bise. I request Shravani Bhosle at Sandeep Kumar. I request Mansi Jadhav to felicitate Vishwajit Veer. I request Komal Sangpal to felicitate Mrs. Jyoti Mandle. I request Nishant Shinde to felicitate Mrs. Ashwini Kakade. On behalf of the institution and all our alumni, extend our heartfelt gratitude to our esteemed faculty members. Your teachings continue to inspire generations, and today's gesture is a small expression of our deep respect and appreciation. Thank you to our alumni for making this moment so special and memorable. Now, we proceed to special moment of today's program by felicitating our distinguished alumni by our esteemed dignitaries. Our alumni are true ambassadors of this institution. Their achievements and contributions bring honor and pride to our alma mater. I respectfully invite our dignitaries, honorable dignitaries, to felicitate distinguished alumni with a token of appreciation. Please, I request all the dignitaries to felicitate the distinguished alumni. Dr. Snehal Masurgar, ma'am.
डिस्टिंग्विश लागते त्याला
First batch. Dr. Jayashree Nanore, ma'am. Dr. Abhay Ghatke, sir. Miss Shivani Guzar. Shivani Guzar. Tejasri Bhagat. Reshma Bhag. Sonika Patil. Shivani Kadam. Sarita Wag. Sarita Wag. Yash Patil. Yash Patil. Shabikta Momin. Gunjan Jadhav. Ravi Jadhav. Meenal Patil. Mohsin Mullah. Sonali Jagtap. Aboli Shinde. Aboli Shinde. Neelam Thorat. Avdut Ghare. Tejasri Varute. Kiran Patil. Nikita Dowry. Saili Jadhav. Saili Jadhav. Payal Bangar. तेजस्विनी कलंत्रे, हर्षदा अहेर, मिसेस स्वप्ना झिंगे, निशांत शिंदे, नमन मिश्रा, तेजश्री देशमुख, प्रणाली पवार, रेवती कंसे, प्राजक्ता घाडगे, श्रावणी भोसले, मनीषा जाधव, कोमल संगपाळ, मानसी जाधव, स्नेहल पाटील, नीलम जगदाळे, Thank you, sir. Let us now give warm round of applause to our distinguished alumni and respected.
So here we are taking a break of 5 minutes. The reason is that Agnihotra is going on in the porch. So we will witness it, and after 5 minutes again we will return back and continue our program. Okay? Agnihotra by Birch, na? Gulikberg. Okay.